Wow. Now I believe I'm audible. Now I believe I'm audible. That's why I was asking you people whether I'm audible or not. So nobody was answering. I thought I'm audible only. So basically for 3 minutes I was just doing, right? Okay, so I believe now I'm perfectly audible. Okay, so first of all, tell me, how are you everybody? Yes, how are you all? How are you all? First of all, tell me. How are you all? Good? Sure? Okay, so let me adjust because the setup is new now. So let me adjust it for a while. So I hope now I'm perfectly audible. Now I'm perfectly audible. Some people are saying we are not good. What happened to you? What happened to you? Don't worry, I'll start the lecture from the starting. Right? Don't worry about that. I'll start the lecture from the starting. Okay, so first of all, welcome to Unacademy NEET English. And initially, I was telling you about this, right? I was telling you about this particular video in which I have completed. I have mentioned the list. It is basically the list of 45 most important chapters. Even if your physics is weak, right? You can follow some of the chapters out of it. You can easily score 90 marks in the physics as well. But yeah, do not miss the PYQs. Do not miss the most important topics there. Okay, and not just for the physics, for the chemistry, for the biology, everything is discussed here in this particular video. So make sure you watch it after class. Secondly, my dear students, here when you look at this, right, today Wasim sir launched this part. This is regarding your test series, right? NEET Crux test series, and in that test series the questions will be there from the NCERT, number one, and basically in all the tests, all the questions will cover your NCERT, will cover your PYQ. So that is going to be amazing for all of you. Okay, that is, what are you talking? Not audible again? How's it possible? How's it possible? Now I believe I'm audible. Now I believe I'm audible. Guys, please, let's not waste the time. Audible now? Some students' voice is clear. Everything is fine. Audible. Yes, I'm audible. Okay, chaliye. So let's start the topic, right? So do not forget about your NCERT Crux series. Okay, chaliye. So let's talk about the topic. So in the last class, bache, we again, as you know, we have completed the most important topics in the last class, right? We have completed few topics from the few topics, but today we will start from the breathing part, right? Basically in the last class we are done with the cell cycle and cell division, right? We have completed the RAS mechanism, urine formation, the counter current mechanism. So these type of basic topics we have already covered. So today, as I said, we will start with the mechanism of breathing. How are we going to start? We are going to start with this particular topic. So students, for the human physiology, right, no doubt your concepts need to be very clear, right? Your concepts, they should be very clear when it comes to the human physiology. But again, I want to suggest you this thing, right? I genuinely want to suggest you this thing, that if let's suppose, right, if let's suppose you are not good in any chapter, then from that particular chapter, at least pick up the most important topic, right? At least pick up the most important topic. At least do not miss that part. Now, when it comes to the mechanism of breathing, how do we need to cover this particular topic? Let me write it here. How do we need to cover this particular topic? Right, for covering this particular topic, we need to take care of few things. And what are that few things, people? What are that few things, people? Number one, you need to know about the muscles. Right, you need to know about the muscles. You need to know about the Boyle's law. Okay, you need to know about the position of the rib cage, position of the diaphragm. These are the things that you need to know. Okay, these are the things that you need to know. Is that clear? Is that clear? Now let's start with the Boyle's law and the basic diffusion thing. Okay, let's start with what? Let's start with the Boyle's law, and let's start with the basic part of this chapter. So when you talk about the diffusion, you know that what is it? What is this diffusion? Right? You know that what is the meaning of diffusion. So when we are saying diffusion, bache, we know that there will be the movement. What will be there? There will be the movement. There will be the movement of gases, right? And how that gases will move from their higher concentration to lower concentration, from their higher concentration to their lower concentration. Basically, not just the gases. In general, when we say that diffusion is the movement of substance from their higher concentration to their lower concentration, from their higher concentration to their lower concentration. So basically, the word that we use here is down the concentration gradient. What is the word that we use here? Down the concentration gradient. That is the term, that is the word that we use, down the concentration gradient. That is the term, that are the words that we use, down the concentration gradient. Yes or no? Yes or no? Kindly keep it in your mind, right? Kindly keep it in your mind. So it is the movement of gases from their higher concentration to, so this is what we know. So now let's say if we want to inhale, we want the oxygen to enter and we want the CO2 to leave, right? Why? Because CO2 is the metabolic waste, right? The CO2 is the metabolic waste. It has to leave and we need the oxygen within. So here we need to remember a law that is known as Boyle's law. Are you getting my point? Right? We need to remember a law. The name of that law is what? The name of the law is Boyle's law. So when you talk about the Boyle's law, you have to remember, if the volume will be high, the pressure is going to be low. If the pressure is high, the volume will be low, right? So pressure, volume high, pressure low, pressure high, volume low. Now you know that, right? If you remember it, very good. If you don't remember it, let me remind you this thing, that when you talk about the lungs, right, you know that they are placed in the thoracic cavity, yes or no? You know that when we talk about the lungs, that lungs are present in the thoracic cavity, number one, right? They are in the thoracic cavity. Now you know that lungs, right, they are even protected, yes or no? They are protected, yes or no? Even they are protected, yes or no? Yes, please tell me. Even they are protected, yes or no? Yes or no? Array, why it's not working? Now it's fine. Take. So when you talk about the lungs, they are the respiratory organ, and we know that when we talk about the lungs, they are present in the rib cage. So dorsally, right, dorsally we have the vertebral column here. And if I have to be specific, so I will say that thoracic vertebrae are present there. What are present there? Thoracic vertebrae. And if I talk about the ventral side, so ventrally what do we have here? We have the sternum. Ventrally sternum is there. Laterally, we talk about the ribs here, right? We talk about the ribs here, and here, right at the bottom part, here we talk about the diaphragm. So this is what we have in the case of lungs. So here I would like to mention one thing, and that is even given in your NCERT. Firstly, dorsally vertebral column, thoracic vertebrae, ventrally sternum will be there, and we have the diaphragm downwards, and laterally ribs are going to protect it. This is what we know. So basically our lungs, they are present in the thoracic cavity. Where are they present? They are present in the thoracic cavity. Where are they present? They are present in the thoracic cavity. This is the first point that we have. Now, because lungs are placed here, but lungs cannot control their volume. Within the lungs, right? Within the lungs, the space within the lungs. What will be the term that we will use for that? We will call it as pulmonary cavity. What are we going to call it? We are going to call it as pulmonary cavity, right? The space within the lungs, what is it? It is the pulmonary cavity. What is it, people? It is the pulmonary cavity. It is the space within the lungs. So lungs cannot control it. Okay, lungs cannot control it. Are you getting my point? Lungs cannot control that. Lungs cannot alter that volume, right? Lungs cannot alter that volume. No. So now what's going to happen? So basically lungs are placed in the thoracic cavity, and they are placed, this anatomical setup is in such a way, right? This anatomical setup is in such a way that if, right, I'm just writing the keywords here. As I said, everything is given in NCERT. You please pay attention on understanding the topic. So if there is any change, if there is any change in the volume of thoracic cavity, if there is any change in the volume of thoracic cavity, there will be the change in pulmonary cavity as well, or basically it will reflect in the pulmonary cavity. there will be a change in the pulmonary cavity as well. There will be a change in the pulmonary cavity as well. So what are we saying? We are saying the simple thing here, right? That this setup is in such a way, if there is any change in the volume of thoracic cavity, if there is any change in the volume of thoracic cavity, there will be the change in the pulmonary cavity as well. The setup is in such a way. Now, why are we talking about this setup? Because we have to understand the Boyle's law here. So just now I told you, if the volume will be high, the pressure will be low. If the pressure will be high, volume will be low. Now, ma'am, how is it related to this particular topic? It's simple. The word that we are using is diffusion. So if we want to inhale, if we want inhalation, if we want what? If we want to inhale, if we want inhalation, right, bache? Basically, we want the air to enter within the lungs, right? We want the air to enter within the lungs. And how is it possible? If outside there will be more pressure and inside if there will be less pressure? Inside if there will be less pressure, yes or no? Outside if there will be more pressure, then inside if it will be less, the air will move from higher to low, right? You know that. It's all about the concentration gradient or pressure gradient. So high to low, the movement will be there. So basically within the lungs, in the pulmonary cavity, if we want to inhale, my dear students, if we want to inhale, so within the lungs, right, in pulmonary cavity, if the volume will be more, if the pressure will be low, don't you think inhalation will occur? Don't you think inhalation will occur? You want to inhale, right? You want to inhale. You want to take the air in. So I want the air in from outside to inside, down the concentration gradient. So outside the pressure, the concentration gradient, your pressure should be high. If inside it will be low, then air will move from high to low. Then air will move from high to low. Then air will move from what? Air will move from high to low. So this is what we want. So if we want to inhale, for inhalation, what is the condition? The volume of pulmonary cavity should be high. The volume of pulmonary cavity should be high. That will result in the low pressure, and that is why the air will enter within. That is why the air will enter within. Now, one more thing. When you are saying air, we know that it is the mixture of gases. We know that it is the mixture of gases. No doubt you want oxygen. We want oxygen. We need this oxygen to oxidize the food. But we need oxygen within, right? Am I clear here? We need oxygen within. We want the oxygen to be there within our body, right? So we want to inhale, and air is the mixture of gases. It doesn't just have oxygen. It is having other gases as well, right? It doesn't just have the oxygen. It is having other gases as well, right? So basically for the air, right, like oxygen is there, carbon dioxide, nitrogen, all are there, okay? So each gas will have the pressure also. Air is basically a mixture of gases. Each gas will also contribute now. Each gas is also having its pressure now. So for that we use the term partial pressure, right? I'm not going to write it, basics they are. For that we use the term partial pressure. What is the term that we use, people? We use the term partial pressure. So partial pressure of oxygen, partial pressure of CO2, that's what you are going to use. So for the inhalation, that's what you need, my dear people. That's what you need. If the volume is high, the pressure has to be low. If the volume is high, the pressure will be low. And that is how the inhalation will occur. So for that high volume, what we have to do? We have to increase the volume of thoracic cavity. So if there will be an increase in the volume of thoracic cavity, yes, bache, dreams unlimited, Nikki, a universal lover, अरे change your name, Prem Gop, right, medic IAS, hi bache. So if I want the volume of thoracic cavity, if I want the volume, more volume here, so of course I have to make changes in the thoracic cavity. That will automatically reflect in the pulmonary cavity, right bache, right bache, right. This is what we want. So for that, what we have to do? We need to talk about the certain muscles. So see, that's how your lungs are placed. That's how they are protected. So this side is ventral side, sternum is there, lateral, and even in the front, you know that some ribs, laterally also ribs, dorsally, you know that vertebral column is there. So what we have, and within this we have the lungs, right? So we need to talk about the muscles here. So now look at the muscles present in between these ribs, right? Look at the muscles. This is first rib, second rib. There are muscles present in between. So the muscles that are present in between, we call them as intercostal muscles. What do we call them, people? We call them as intercostal muscles. What do we call them, people? We call them as intercostal muscles. Intercostal muscles they are, because they are present in between ribs. So just imagine, these are two ribs. Here you have the muscles, right? External Intercostal muscle mane present in between ribs. So if it is present externally, external intercostal muscle, right? If it is present externally, it will be external intercostal muscle, and let's suppose it is present internally, right? Right, internal side is also having something, so internal intercostal muscle. So external, internal, external, internal. The point here is they are present in between the ribs. So this, the first word is intercostal muscles. So we have external intercostal muscle and we have internal intercostal muscle. So let's come to the direct point here. So external intercostal muscles, when they contract, your rib cage moves upward and outward. Firstly, write down these points and then I'll explain it. Upward and downward. Upward and downward. This is about the first muscle. So basically, we are talking about the muscles here. I told you about which muscles? I told you about the intercostal muscles. So I just told you that in between the ribs, you have the intercostal muscle. If they are on external side, external intercostal muscle. If they are on internal side, we have the internal intercostal muscle, right? And other than that, we have the peripheral muscles of diaphragm, or you can simply say diaphragm muscles. You can simply say diaphragm muscles, right? Or we can also say these are the peripheral muscles of diaphragm, or we can also say these are the peripheral muscles of diaphragm. These are the peripheral muscles of diaphragm. So remember, right? Remember, during, when we are not inhaling, your diaphragm is dome shaped. Your diaphragm is dome shaped. It is relaxed, it is dome shaped. But when we will inhale, the diaphragm will become flat. So when diaphragm muscles, when these muscles contract, diaphragm becomes flat. Diaphragm will become flat. Now, what we need to understand from this particular portion, our understanding is quite simple here, bache. Let's suppose if this is the, right, I know the diagram is pathetic, but let's suppose if this is the diagram. Yeah, sorry. If let's suppose this is the thing. Now, if, right, normally, right, when you are not inhaling, when there is exhalation, when you are not inhaling, when there is exhalation. So during exhalation, your diaphragm will be like this, right? Your diaphragm will be like this, right? Posteriorly diaphragm is present now. Diaphragm is like this. So basically during exhalation, diaphragm muscles are relaxed. They are not contracted, right? Muscles are not contracted. They are relaxed now. They are not contracted. So because these muscles, they are not contracted, the diaphragm here is dome shaped. It is dome shaped. Now during inhalation, when there is inhalation, when there is what, people? When there is inhalation, then what is happening, right? You know the diaphragm muscles. I'm using the short form here. Diaphragm muscles will contract. So diaphragm will become flat. So it will be something like this. Something like this. So don't you think that will increase the volume of, right? So volume will increase towards anterior posterior axis. Volume will increase towards anterior posterior axis. Volume will increase towards anterior posterior axis. Am I clear? Yes, am I clear? Done. Number one. Now, when you are talking about the external intercostal muscles, now, when that muscles will contract, I told you already, your rib cage, right? Your rib cage will go upward and outward. Upward and outward, right? Let's suppose if these are the ribs, these are the muscles present in between, right? Now they are contracting. So here, you know that I explain it in one way. You have to remember this thing. This is your first dance step. This is your second dance step. This is your first dance step. This is your second dance step. Do it all together. So it is showing that diaphragm muscles, when they will contract, diaphragm will become flat. So the volume towards the towards the anterior posterior axis will increase. Now when you are talking about the muscles, external intercostal muscles, when that external intercostal muscles, when they contract, the rib cage will move outward, upward, outward, upward, outward, upward. Am I clear here? So rib cage will move outward, upward, outward, upward, outward, upward. That's what we have to remember. Okay, so these two muscles, because they contract during, right, the inspiration, so we call them as inspiratory muscles. What do we call them? Inspiratory muscles or inhalation muscles. Why? Because they contract during inspiration. Because they contract during inspiration. Are you getting my point? Because they contract during inspiration. So diaphragm muscles and external intercostal muscles, they contract. Diaphragm muscles or external intercostal muscles, they contract. So this is about the inhalation, right? Now you know that, so there should be an increase in the volume, right? There should be an increase in the volume. So that's how the muscles will contract. Now what's going to happen, right, in the case of exhalation? So what will happen? What will happen? What do we need? We need to throw out the CO2. We need to exhale out the CO2. We need to pass the CO2 outside, right? We need to pass the CO2 outside. That is what we need. Yes, people, that is what we need. Yes or no? That is what we need. Hannah, this is what is our requirement. So here what is going to happen? Of course, your external intercostal muscles, your diaphragm muscles, they will relax. So when they will relax, bache, when they will relax, of course, the rib cage will move downward and, right, it will become, it is basically moving outward or upward now. So it will become downward and inward. Right, bache? Right, bache? So when your external intercostal muscles relax, so what will be the position of the rib cage? So your rib cage, bache, right, rib cage, inward and downward. And diaphragm, so here diaphragm will become dome shaped. Okay, dome shaped. So remember one more thing here, when you are talking about the inspiration, inhalation, right, when you are talking about the inspiration, inhalation, right, bache, remember one thing, your inhalation is an active process. So when we say active process, means it will use energy. Am I right? So when we say yes. So when we say what? When we say inhalation, what kind of process it is, people? What kind of process it is? It is going to be your active process. Are we clear here? Yes, please. Are we clear here? So what kind of process it is? What kind of process it is? It is going to be the active process. What kind of process it is? It is going to be the active process. Anybody having any doubt? Yes. Anybody having any doubt? Sure, sure. Right? Are you sure? Pakka? Are you sure? So, inhalation active process for 0.2 seconds. Right, bache? When you talk about the exhalation, the conditions are opposite. Now, here volume has to go down, pressure has to increase. Right? Then only the CO2 will go out. So, it is 0.3 second and it is a passive process. What kind of process it is, people? It's a passive process. What kind of process it is? What kind of process it is? It is the passive process. So, I believe this portion is clear. I believe this portion is clear. Right? I hope it is clear. Okay, so now there is one more thing that is known as, right, what is that? That is basically the, yes, forceful expiration. What is that? That is basically the forceful expiration. So, bache, in forceful expiration, forcibly, right, forcibly you are excreting, forcibly you are exhaling. What are you doing? Forcefully you are exhaling. So now what's going to happen? Right, overall, right, forceful expiration, it is an active process. Remember this thing, it is not passive, it is an active process. What kind of process it is? So your inspiratory muscles and all will so basically when there is the forceful expiration, then in that case what's going to happen? So in forceful expiration, active process it is, your internal intercostal muscles will also contract. We have to reduce the volume further. Is that clear? Is that clear? Anybody having any doubt? So movement clear, right? So when there is the expiration, you tell me. And yes, we can use this additional muscle, stomach muscles are also used, right? Right? So now you tell me, I'm asking you this question. The position clear, inhalation clear, exhalation clear. So now you know which part will be contracted in inhalation, which part will be relaxed in exhalation. This is what we have already discussed. So when we inhale, what will be the lung pressure? What is going to be the lung pressure? Intrapulmonary pressure. What is going to be the intrapulmonary pressure when we are inhaling, Teju? When we are inhaling, what about that intrapulmonary pressure? Tell me quickly. When we inhale, so what is the status of that intrapulmonary pressure? Quick, tell me. Yes, please. What should be? Hanji, intrapulmonary pressure within the lungs, the pressure has to be low. So inhalation mein intrapulmonary pressure or pressure within the lungs, it's all same. So that intrapulmonary pressure and the pressure within the lungs. So that intrapulmonary pressure, what about that? So that pressure will be negative. It is decreased. So we can also call it as negative intrapulmonary pressure. So look at the topic here. Let's have a quick revision of it. So when we revise it quickly, so you can see inspiration during which atmospheric air is drawn in, expiration by which the alveolar air is released out. Okay? Alveolar air is released out. So the movement of air into and out of the lung is carried out by creating a pressure gradient between the lungs. Okay? So inspiration will occur if the pressure within the lungs, that is intrapulmonary pressure, is less than the atmospheric pressure. So it means there has to be the negative pressure in the lungs with respect to the atmospheric pressure. So do we have any doubt here? 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 No? Done? So then expiration mein the intrapulmonary pressure is higher than the atmospheric pressure, right? So we have the external intercostal muscles between the ribs and right, they will help in maintaining such gradient. Take, so the volume will increase. I told you already about it, right? So this is the quick revision part. If anybody is interested in revising it in more detail, you can watch my one shots. So diaphragm is contracted here. So when diaphragm is contracted, it will become flat. It will not be dome shaped anymore. So your rib cage, ribs and sternum, they are raised. Do we need to pay attention here? Definitely we have to, right? I'm sorry. So we are not allowed to miss anything here. Anything means anything, right? And especially the diagrams, especially the diagrams and the tables. Okay, so now air is expelled from the lungs, so volume of thorax will decrease. Here the volume of thorax will increase. So if volume of thorax is increasing, that will in return will also increase the volume of pulmonary cavity. That's what you need to keep in your mind. So ribs and sternum return to their original position. Keep it in your mind. So diaphragm is relaxed, it is arched upwards, means it will become dome shaped. Done. Next is the exchange of gases. Again, a very simple topic and the important one, right? So exchange of gases will occur by a diffusion. We know that there should be the difference in the partial pressure. So here, what is important? The table that is given regarding the partial pressure, na? This is the figure, but there is a table also. Just keep it in your mind, the difference in partial pressure. That is also important. So when you look at this alveolar air, what is the partial pressure in alveolar air? This is a PO2. So what is the term that I have used? Partial pressure of oxygen. So because air is a mixture of gases. Air is a mixture of gases. Just let's suppose you all friends are like that, today we want to eat pizza and let's contribute, right? And you are contributing 100 rupees each. So you got a pizza, right? No, so obviously you all have the right, na? Right? You have contributed the equal amount of money. So you all have the right on that pizza, right, bache? Same is the case here. So when you are talking about the air, which is a mixture of gases, there are many gases there. So each gas is contributing there. Each gas is having a specific pressure there that will make the total pressure of the air. So for that, we use the word partial pressure. For that, we use the word partial pressure. Is that clear? Is that clear? That's what you have to remember. So alveolar air, partial pressure of oxygen is 104 mmHg, for CO2 40 mmHg. So can we get question from this part? Definitely it's a yes. Now you know that the exchange of gases, diffusion has to be done, right? So you know that blood capillaries are innervating this alveoli. So here this exchange of gases will be there in between the alveoli and blood vessels. So we call it as external respiration, right? And why is it possible? Due to the difference in the partial pressure. So here in that blood vessel, the partial pressure of CO2 is high, and here it is, and oxygen is low, and in the alveoli it is high. So of course exchange will be there. Now you know that that's how the exchange will be. And then rest we know that it is the heart which is the pumping organ. It is the heart which is the pumping organ, which has to pump the blood. So in the case of heart also, you know, one trick is there, LOD. Like left side will have oxygenated blood, right side will have deoxygenated blood. Right side will have deoxygenated blood. So basically left side will pass that blood to the complete body, right? And left side, right side will take it to the lungs. So you know that we have the exception here, pulmonary artery, right? That's how we revise the pictures. We have to recall all the points related to that topic, right? That's the best way to revise, right? That's the best way to revise that particular part. So what is the word that we have? We have pulmonary artery. So pulmonary artery, they will carry the deoxygenated blood, right? Because it is an exception. Why? Because it is an exception. It is the one that will carry the deoxygenated blood. So it will take it from the right ventricle to the lungs, because right side is having deoxygenated blood. So it will be pulmonary artery. Then pulmonary vein, you know that it carries the oxygenated blood. It will carry it to the left auricle, then to the left ventricle. Then which is systemic artery, right? Systemic artery will carry it to the body parts. And here it is the systemic vein that will bring back the deoxygenated blood to the right auricle. And it is the pulmonary aorta or artery that will carry it to the lungs. Okay, that will carry it to the lungs, and here it is the systemic artery. So then the next exchange will be there in between the blood capillaries, right, and in between the blood and the tissue. And here this is known as internal respiration, right? So here you will see that within the tissue, the partial pressure of carbon dioxide is 40 mmHg, and the partial pressure of oxygen is this. So there is a table given in NCERT that is also very, very, very important, right? That is also very, very, very important. And I'm in the month of April, so we have the one shots, but I'm not talking about that. I'll post the short videos in which I will cover all the diagram and all the tables from particular units. Just we have 5 units, so we'll complete it like this. Take units, I'm saying, not the chapters. So this is how the exchange is. Now this topic, right? Meanwhile, let's complete it here. You have the alveolar cavity. You have the alveolar cavity. You know that the diffusion will occur. So what type of epithelium do we have here? We have the simple mane single cell layered, simple squamous epithelium. What do we have there? We have the simple squamous epithelium. What do we have there? We have the simple squamous epithelium. Simple squamous epithelium, and it is for the alveoli. And you know that they will lie on what? Basement membrane. You know, epithelial tissue. Epithelial tissue. This is the epithelial tissue. It will lie on what? It will lie on basement membrane, right? So when you talk about this basement membrane, when you talk about that, it is basically acellular, not made up of any type of cells. It is basically acellular, not made up of any type of cell. Take. And now the blood capillary. So blood capillary endothelium is there. They also lie on the basement membrane. They also lie on the basement membrane. So that is how the exchange will occur here. That is how the exchange will occur here. So these 3 layers. Number 1, the simple squamous epithelium of alveoli, right? The endothelial of blood capillary, and basement membrane present in 2. Take. So that's how the exchange will occur. Okay. So remember that, bache, solubility of carbon dioxide is 20 to 25 times higher than that of the oxygen. These are the important lines that I'm also highlighting, and they all are the PYQ. Now, the other thing that we need to focus is transport of gases. What we need to understand, people? We need to understand the transport of gases. What do we need to understand? We need to understand the transport of gases. Okay. Okay. So let's start with the oxygen. You know that 97% of oxygen, right? Database questions are also there. They'll form oxyhemoglobin. What will they do? They will form oxyhemoglobin. So what's the first point here? What is this hemoglobin? Hemoglobin, right? What is this hemoglobin? Let me write it here only, HB. We know that it is our respiratory pigment. What's that, bache? It is our respiratory pigment. What is it, bache? Yes, everybody here in the class. The hemoglobin is what? The hemoglobin is the respiratory pigment. What is this hemoglobin? What is this hemoglobin? It is the respiratory pigment. What is it? It is the respiratory pigment. And we know that it's a quaternary protein. It's a quaternary protein. Remember, it's a quaternary protein. It is having 2 alpha chains and 2 beta chains. It is having 2 alpha chains and 2 beta chains. That's what we have. It is having 2 alpha, 2 beta chains, right? 2 alpha, 2 beta chains. That's what we have when we talk about what? When we talk about what? When we talk about the hemoglobin, adult hemoglobin. Now quaternary protein it is. So you can imagine the structure like this, right? Like these polypeptide chains look like the woolen balls now. So that is what we have. So when it is, the word is hemoglobin, HB, hemoglobin. So when you talk about this hemoglobin, students, so heme is the pigment, globin is the protein. Heme is the pigment, globin is the protein. Yes, Nikki. What do we have here, bache? Heme is the pigment and globin is the protein. So anybody having any doubt here? Yes. Anybody having any doubt here? Yes. Heme is the pigment, globin is the protein. So you have 2 alpha, 2 beta. So this part here is your heme, rest is globin. So remember here, which metal ion is there? Iron is present. So it is in Fe2 plus state. That is important, right? We know that iron is required here. So it is in Fe2 plus state, right, bache? So this iron is in Fe2 plus state, right? This iron is in Fe2 plus state. Is that clear? Okay. Are you sure? Pakka? Done? Done? Yes? Sure? Okay, chali. So iron is in Fe. Iron is in Fe2 plus state. So you know that each iron. So basically we have hemoglobin, quaternary protein. So we have Fe2 positive, right? And each of it will join with the oxygen, right? So basically, if you talk about one hemoglobin molecule, it will bind with 4 oxygen molecules. If you talk about one hemoglobin, it will bind with 4 oxygen molecules. So how do we write it? We write the equation like this. Yes or no? Yes or no? Have you seen that? That is how we write it. Yes or no? Yes or no? That's how we write it. Yes or no? Yes or no? Yes, please. So that is how exactly. Yes. So Hb4, 4O2, and this is what we are going to get, right? So basically we are talking about the oxygen transport. We know that 97% of the oxygen will be transported, right, by the formation of oxyhemoglobin. So who's going to help that, right? Hemoglobin, where is it present? Within the RBC. We know that it is the respiratory pigment, and because of that blood is red in color. So here also 97% of oxygen is transported by RBCs in blood, and 3% will be there in the dissolved state. So first of all, tell me this information, is it clear? Because now, right, this information is clear. This information is clear. Sure. Sure. Chaliye. So what is the next part that we have? So this is the equation. Basically, hemoglobin will join with 4 oxygen molecule. Why? I told you, right? I told you about the iron and everything, right? That is how oxyhemoglobin will form. Now, bache, the point here is that basically when there is the formation of oxyhemoglobin, we use the word oxygenation here. What is the word that we use here? Oxygenation. Mane addition of oxygen. So we should not use the word oxidation. That is having the different meaning. Right, that is having the different meaning. We are using the word oxygenation, that is addition of oxygen. So basically the oxygen will be added to the right hemoglobin. That's the point. Now when you talk about this binding, right, this binding is temporary and reversible. Matlab this binding of oxygen and hemoglobin, it is temporary and reversible. I'm just writing the important line here. It is temporary and reversible. What is it? What is it? It is temporary and reversible. It is temporary and reversible. Are you getting my point? Are you getting my point? Why temporary and why reversible? It's simple. We have to add the oxygen into the hemoglobin because hemoglobin is a pigment that will help in the transport of oxygen. Just say myoglobin. It is in the muscle. It stores the oxygen. This hemoglobin, it helps in the transport of oxygen. It's just like a delivery boy, delivery person, right? Let's suppose you have ordered something expensive, a phone, Android phone, iPhone, whatever you like. Now, the delivery person has to carry it, right? In that time, it is his responsibility to hold it properly, right? To keep it safe, and then that person will give it to you. Okay. So if let's suppose, so that is a basically kind of temporary relationship in between that, phone and that person, right? They cannot keep it permanently. They just need to keep it for some time and then they have to deliver it to you. Same is the case here. Hemoglobin is the pigment that is basically for the transport of gases. So if it is going to pick up the oxygen, this binding has to be temporary and reversible only, because if it will be irreversible, right bache, then our cells will not get the oxygen. Done? Done? So it will be temporary and reversible. It will be temporary and reversible. Are you getting my point? It will be what? It will be temporary and reversible. Is that clear? Is that clear? Right. So it is temporary and reversible. Now, for this oxygenation, what are the conditions? Okay, we got it. That oxygen will be transported by this hemoglobin by the formation of oxyhemoglobin. So first thing is the high partial pressure of oxygen. Remember this thing in the case of hemoglobin. If there will be the high partial pressure of oxygen, it will bind with oxygen. If there will be the high partial pressure of carbon dioxide, it will bind with carbon dioxide. So it's all the game of partial pressure here. It's all the game of partial pressure here. Right, bache? Right. Yes? Yes? Sure? Okay, chaliye. So high partial pressure of oxygen. Second is low partial pressure of CO2. Right. Third is what? High partial pressure of CO2, high partial pressure of oxygen, low partial pressure of CO2. So in that case, there will be high pH. When I'm saying high pH means low H positive concentration. And of course, in that case, the temperature will also be, anybody here in the class? What about the temperature? What about the temperature? What about the temperature? Because all that factors, they affect the oxyhemoglobin formation. So can you please tell me what about the temperature? Anybody in the class? Exactly. So the temperature will also be low, right? It will also be low. Is that clear? Is that clear? These are the conditions for oxygenation. If these conditions are available, oxygen will form. Hemoglobin will bind with oxygen, right? If these conditions are available, hemoglobin will bind with oxygen. This point clear? Now, remember, in 100 ml of blood, you know that on an average, in 100 ml of blood, 14 to 16, 14 to 15 gram of hemoglobin is present. Do you know that? 14 to 15 gram of hemoglobin is present. Do you know that? Yes, please. 14 to 50 gram of hemoglobin is present on an average. 14 to 15 or 14 to 16 it can be, or 12 to 14. Because in the case of girls, in the case of females, it is less. Yes, it is less. So what should I write? Should I write 14 to 15 or should I write 12 to 15 according to you? Yes? Done? What should I write? I'm saying 100 ml of blood. Bache, why are you writing 5 ml and all? You didn't understand my question? You didn't understand my question. I'm saying 100 ml of blood will have 14, right? You can say that 12 to 15 gram, yeah, 15, like it is in that range, because in the girls, right, in the women, the hemoglobin is usually less, right? So 100 ml of blood we are talking about, and this much of hemoglobin is present. And you know, 1 gram of hemoglobin, 1 gram of hemoglobin can bind with 1.34 ml of O2. Do you know that? 1.34 ml of O2. Do you know that? So 1 gram of hemoglobin can bind, or if it can bind, so obviously it can transport, right? So 1 gram of hemoglobin can transport 1.34 ml of oxygen. Up to this part all clear? Up to this part all clear? So just imagine if in 100 ml of blood, if we have 15 gram of hemoglobin, what you have to do? 15 into 1.34. So somewhere it is around 19.4 ml. So can I say that somewhere around 20 ml of oxygen will be transported by, right, 100 ml of blood? Can I say so? Can I say so? You tell me, can I say so? Can I say so? Quick, calculate it and tell me. Calculate it and tell me. Right, it is not, it is 15 into, if I'm picking up 15, it will be near about 20 only. Directly, let's write 20. So it is around 20 ml of oxygen. Right? So what am I saying? Firstly, I told you that yes, hemoglobin will bind with oxygen. It will form oxyhemoglobin. This binding has to be temporary and reversible. I told you, I have given you the reason as well. Second thing that I'm saying is that for oxygenation, for addition of oxygen, for the formation of oxyhemoglobin, these are the conditions that we need. These are the conditions that we need. It should be on your fingertips. It's obvious. If there is high partial pressure of oxygen, the partial pressure of CO2 has to be low. If low CO2, matlab metabolic reactions are not going on, metabolism is less, so low temperature condition, high pH, that is low H positive concentration will be there. Second, on second side, basically here, what am I writing? I'm saying 100 ml of blood. It is having around 12 to 15 gram of hemoglobin. 1 gram of hemoglobin will transport, 1 gram of hemoglobin will transport 1.34 ml of oxygen. Right, 1.34 ml of oxygen. Right, bache, 15 gram will give 15 into 1.34 ml. Right, 15 into 1.34 ml, and that will be 20 ml. That will be 20 ml. So in short, I am saying that 100 ml of blood, 100 ml of blood will carry what? 20 ml of oxygen. Right, so it is about the oxygenated blood. It is about what, bache? It is about the oxygenated blood. Is that clear? Is that clear? Right? Is that clear? Is that clear? Matlab, this is what it will carry. This is what it will transport. Right? This part clear? This part clear? Okay. So when there are normal physiological condition, so under normal physiological condition, under what? Under normal physiological conditions. What is the point here? See, this is for the oxygenated blood. This is for what? This is for the oxygenated blood. This is for what? This is for the oxygenated blood. So under normal physiological condition, 5 ml of oxygen will be delivered, right, by this hemoglobin to the respective tissue, right? It will be delivered, because when we check, just say this is the 100 ml of oxygenated blood, this is the amount of oxygen. If you will check 100 ml of deoxygenated blood, so it will be around 15. So basically we can say ki 5 ml of oxygen, 5 ml of oxygen is delivered. Right, 5 ml of oxygen is delivered. Right, 5 ml of oxygen is delivered. This is what you need to keep in your mind. So up to this part all clear? Up to this part all clear? right? So when we talk about the formation of this oxyhemoglobin, it is the oxygenation. When we talk about the formation of the oxyhemoglobin, what is the word that we use, bache? It is the oxygenation. Do you have any doubt here? Do you have any doubt here? So second thing that we have to discuss here is the, jaise ye dekho, even here, right, they are talking about the oxyhemoglobin. I told you already, right? So each hemoglobin molecule, they can carry what? Each hemoglobin molecule, they can carry a maximum of 4 molecules of oxygen. So binding of oxygen with hemoglobin is primarily related to the partial pressure of oxygen, right? So you know that partial pressure of CO2, H positive concentration, temperature, they all are the factors which will interfere with this binding. So now a sigmoid curve is obtained where percentage saturation of hemoglobin with oxygen is plotted. Take. So a sigmoid curve is obtained. So basically we are talking about the oxygen dissociation curve, या oxyhemoglobin dissociation curve. So what is there? First of all, it is S-shaped. When am I saying S-shaped? It is sigmoid. So here we need to remember some shifts. Take. So it is a graph which is plotted in between the oxygen concentration and percentage saturation of hemoglobin. Right? It is a graph that is plotted in between the concentration of oxygen, right? Between the oxygen concentration and percentage saturation of hemoglobin with this curve. This is the oxygen hemoglobin dissociation curve, right? So when we use this curve, it is very helpful for study. It is useful in studying the effect of factors like partial pressure of CO2, H positive concentration on binding of hemoglobin with oxygen. Is that clear? Now here in this curve, we need to remember few things. And what are that few things? We need to remember the shifts. And for the shifts, we have the trick. So what is the word that we have? We have the word LAD. So L stands for left, A stands for association. L stands for left, A stands for association, R stands for right, D stands for dissociation. They said we have one trick as LOD, another trick is LAD, right? We have one trick as LOD, one trick as LOD in the case of heart. Here we have what? Here we have LAD. Here we have LAD. Is that clear? Sure. Sure. So the trick is LAD. So here, this LAD, right, left, right. So basically, LAD is the word, right? Basically, LAD is the word. Left is association, right is dissociation. Left is association, right is dissociation. So left side is dissociation. Association matlab means oxygen and hemoglobin. They'll associate, means they will bind. They'll bind means oxyhemoglobin will form. That's the way to remember the things, right? It means oxyhemoglobin will form. Yes or no? Yes or no? Yes or no? So I am saying that oxygen and hemoglobin, left side means LAD is the trick. So left side means association. So it simply means oxygen and hemoglobin. What will they do? They will bind, right? If they will bind, what will they form? Oxyhemoglobin. So this is for the left shift. So you know that here oxygenation will be there. And when it is the oxygenation, you know the conditions, right? You people know the conditions. Yes or no? You know the conditions that there has to be high partial pressure of oxygen. There has to be low partial pressure of CO2. There has to be high H positive concentration. What should be there? High H positive concentration. Low H positive concentration or high pH, right? Here there should be high pH, right? That means there will be high pH means low H positive concentration and the temperature will be low. Are we clear here? Now, right side is about the dissociation. Means oxygen and hemoglobin, they have to separate. Is it important? Is their separation important? Yes. This is important because if oxyhemoglobin, if they will not separate from each other, right? If they will not separate from each other, my dear people, then how our body cells will get the oxygen. So here this breakup is important, right? Because if oxygen and hemoglobin, if they will have breakup, then only our oxygen will go to the cells, to the tissue, and that's what we want. So this breakup is very important for the transport of oxygen. Remember, hemoglobin is there for the transport. Hemoglobin is there for the transport of oxygen. Hemoglobin is there for what? Hemoglobin is there for the transport of oxygen, right? But it is not the pigment, oxygen storing pigment. It is there for the transport of oxygen. This is the word that you need to keep in your mind. This is the word that you need to keep in your mind. Number one. Now we know that, right, that 100 ml of blood is having 15 gram of hemoglobin, and each gram of hemoglobin can transport 1.34 ml of oxygen. So 100 ml will have 20, right? 20 ml of oxygen approximately. So when you check the deoxygenated blood, the concentration is 15. So we will subtract it. 5 ml of oxygen is delivered. That's what we have covered. Now we are talking about this large thing. We are talking about this oxygen hemoglobin dissociation curve. We are talking about the shift. So left shift, if it means association, so it's simply, right, if it means association, so it's simple, my dear people, it's simple. What is going to happen here? Oxygenation, these are the conditions. Right side, dissociation means breakup. So from the breakup word, you have to remember Bohr's effect. From the breakup word, you have to remember Bohr's effect. What you have to remember? You have to remember Bohr's effect. So the opposite conditions will be there. What are that opposite conditions? There will be high partial pressure of carbon dioxide. There will be low partial pressure of oxygen. There will be high H positive concentration. It means there will be low pH and there will be high temperature. These are the conditions. So where will you see such conditions where high partial pressure of CO2 is there? Right, bache, you will see such conditions in the tissue. In the tissue. And that is why, because there will be the breakup, because these are the conditions, so oxygen will be transported to the tissue. So if in the paper there is a question, which effect will help in the transport of oxygen, the answer is Bohr's effect, right? And what about this left shift? Where will you see the left shift? Of course, L for left, L for lungs. Why lungs? Because in the lungs, high partial pressure of oxygen is there. Right? And this effect is actually known as Haldane's effect. This patch-up effect is also known as Haldane's effect. Clear? Haldane's effect. It is Haldane's effect. It is Haldane's effect. So left shift, these are the conditions. Right shift. So in NCERT it is not mentioned, but here they are talking about 2,3-diphosphoglycerate, which is even low in this particular case. Right? Diphosphoglycerate or bisphosphoglycerate it is. So any doubt? Just have a look. Any doubt? Just have a look. Any doubt? Done. So this is what we have. So in the NCERT, they have just given one line, every 100 ml of oxygenated blood can deliver around 5 ml of oxygen. And I have explained you why. Any doubt? Sure? Are you sure? Yes, please. Chaliye, very good. So now the transport of CO2, let's discuss about that. So we know that, right? We know that maximum CO2 will be transported in the form of bicarbonates. It's a PYQ. It's a PYQ. Ye dekho. It's a PYQ. Nearly 20 to 25% of the CO2 is transported by RBCs. 20 to 25% of the CO2 is transported by RBC, whereas 70% of it will be carried as bicarbonate, HCO3 negative. Bicarbonate, HCO3 negative, and 7% of CO2 is carried in the dissolved state through plasma. So is it important? 110%. 110%. It's very important. Now when it comes to the transport of CO2, the solubility of CO2 is more. It is 20 to 25 times. So solubility of carbon dioxide is 20 to 25 times more than that of, it is more than that of 20 to 25 times more than that of oxygen. So as I said, it will form bicarbonate. It will be there in dissolved state as well. And right, and ye dekho, CO2 is carried by hemoglobin as carbaminohemoglobin, about 20 to 25%. So majority of it will be transported in the form of bicarbonates. Then you know that bicarbaminohemoglobin, baki in the dissolved state, 70%, right? Something like that. No, something like that, nahi, exactly like that, because the question will come na. So here we cannot do this something wala thing. Yes, we have to remember this. See, we have to remember this. Okay. So 70% bicarbonate, 7% will be dissolved state से plasma के through. Now, what we have to remember here, see, I'm CO2 plus hemoglobin. One thing is for sure, if we want to bind them together, right, then of course the partial pressure of carbon dioxide should be high. It means partial pressure of oxygen will be low. It means high H positive ion concentration and high temperature, right? That is something you have to keep in your mind. But the thing that I want to highlight here is this one. When carbon dioxide and hemoglobin, they bind, the word is carbaminohemoglobin. When carbon dioxide and hemoglobin, when they bind, the word is carbaminohemoglobin. What is the word that we have? Carbaminohemoglobin. So same way, my dear students, carbon monoxide can also bind with hemoglobin. So when carbon monoxide binds with hemoglobin, that is carboxyhemoglobin. That is carboxyhemoglobin. That is carboxyhemoglobin. Is that clear? That is what? That is carboxyhemoglobin. Am I clear? That is carboxyhemoglobin. Is that clear? Is that clear? Are you sure? Pakka? Any doubt? Any doubt? None, bache? Okay. Done. So this is carboxyhemoglobin. So I have seen students making the mistake here. Carbon dioxide plus hemoglobin is carbaminohemoglobin, but when it is carbon monoxide plus hemoglobin, it will be carboxyhemoglobin, right? It is what? It is carboxyhemoglobin. What is it, bache? It is carboxyhemoglobin. Are you getting my point? Are you getting my point? So that is something you need to keep in your mind, right? Number one. Take. So again, as I said, this binding is also related to the partial pressure. So are we going to discuss it in this way? No, right? No. We have to discuss it in a little different way. But of course, if carbon dioxide has to bind with hemoglobin, one thing is clear, high partial pressure of carbon dioxide, then there will be low partial pressure of oxygen, right? And the conditions accordingly. Now, we have to understand one thing. We have to talk about the fastest enzyme here, right? Let's suppose this is the RBC. In our body, whenever, and in the surrounding, there is plasma, because we know that when we talk about the blood, we have formed elements and plasma. We have formed elements and plasma. What do we have, people? We have formed elements and plasma. Is that clear? Is that clear? Is that clear? Now, here carbon dioxide and water, they will bind, and it will result in the formation of H2CO3, that is carbonic acid. That is carbonic acid. जैसे C, your CO2 plus water. This is your RBC. In the surrounding, we have plasma, right? Carbon dioxide plus water. It is carbonic acid. It is carbonic acid. It is H2CO3. What do we call it? We call it as carbonic acid. This reaction is important, right? So here, when you talk about this reaction, there is an enzyme that is going to catalyze it, CAH. What is this CAH? This CAH stands for carbonic anhydrase. This CAH stands for carbonic anhydrase. And let me tell you, students, it is the fastest enzyme. What's that? It is the fastest enzyme. What's that? It is the fastest enzyme. Now, this carbonic acid, it's a weak acid. It will dissociate. Again, the same enzyme will dissociate it. And we will get bicarbonate plus H positive ion. What do we get, bache? We will get H positive and bicarbonate ion. We will get H positive and bicarbonate ion. Is that clear? Is that clear? Yes or no? Yes or no? Now, you know that it is in the RBC. In the RBC, you have the hemoglobin. In the RBC, you know that you have the hemoglobin. Yes or no? Yes or no? Now, this H positive which is released here, it will bind with the hemoglobin here and it will result in the formation of hemoglobinic acid, right? So, dekho, I just told you, when there is the, right, high partial pressure of CO2, when this reaction is taking place, when there is the high partial pressure of CO2, when there is the high, bache, is my voice is not clear? Gayatri, seriously? Guys, is there any issue with the voice? Hanji, is there any issue with the voice? I believe it's clear. Preeti, Gayatri, is it clear or not? Hello? Sometimes, matlab, why? Sometimes as in, like, what's the issue? What's the issue? Clear? Voice is breaking. Chalo, let's wait. Chalo, let's wait for some time, then we will see. Then we will see. Okay. Now, this H positive, as I said, what's going to happen to this H positive, my dear people? So when you talk about this H positive, it will bind with the hemoglobin and what will you get? Hemoglobinic acid. What will you get? Hemoglobinic acid. So I told you now that when there is the high partial pressure of CO2, there will be low pH, that is high H positive concentration. Now you can understand why. Now you can understand why. Are you getting my point? Now you people can understand that why is it so. Am I clear? Why is it so? Am I clear? Am I clear? Yes or no? Yes or no? So that's how we write this hemoglobinic acid. That's how we write this hemoglobinic acid. Okay, that's how we write this hemoglobinic acid. Now, what about these bicarbonates ion? These bicarbonates are ions are right. Dekho, we are talking about the enzyme carbonic anhydrase. Bache, it is present in RBC as well as in plasma. As well as in plasma. It is present in RBC as well as in plasma. It is present in RBC as well as in plasma. But in plasma, its quantity is negligible. Its quantity is negligible. Its quantity is negligible. Its quantity is negligible. Now, this bicarbonate is like, I just want to go out. I don't want to stay here. I want to go out. This bicarbonate, right? This bicarbonate is like, I don't want to stay here. I want to go outside. If we will send this bicarbonate outside, right? If we will send this bicarbonate outside, then what will happen? Ionic disturbance can be there. So, bicarbonate is like, I am too much here. Outside, I am very less. Throw me out. Theek hai ji. We are going to throw it out. But we have to balance out the things as well. So, chloride will enter. So, basically here, there is one more thing that is happening. That is the chloride shift. That is the chloride shift. What is happening here, bache? Chloride shift. That is also known as hamburger phenomena. That is also known as hamburger phenomena. So there is the chloride shift, and it is also known as hamburger phenomena. So what is happening here, my dear people? The simple thing here is that bicarbonate ion is going out, bicarbonate going out in the plasma, and chloride is entering, right? And the chloride is entering, right? And the chloride is entering. So this is the chloride shift, yeah, hamburger phenomena. So basically we have to maintain that balance. If bicarbonate is going out, chloride is also entering, so that charge will be balanced, so that charge will be balanced. So this point clear, this point clear, right bache? This point clear, yes or no? Yes or no? Now this bicarbonate which is diffused out, so it is diffused out as per concentration gradient. It is more here, it is less here, it is go out. It will join with sodium ions, right? And it will form NaHCO3, sodium bicarbonate. What will it form, bache? It will form sodium bicarbonate. What will it form? It will form sodium bicarbonate. What will it form, my dear people? It will form sodium bicarbonate. What will it do, people? What will it do, people? It will form sodium bicarbonate. It will form sodium bicarbonate. Is that clear? Is that clear? Are you sure? Right? So that is how it will be transported. That is how it will be transported. So this part clear. So 70% of the CO2 will be transported by making the bicarbonate ion. So I believe this part is clear to everybody, right? Right. So that's how this transfer will be. Look at this picture here, right? Look at this picture here. So that's going to happen. So of course, right, the chloride shift is written here, and this is happening. Where is it happening? Of course, tissue part. Of course, tissue part. Why? Right, in the tissue this has to happen now, right? Because there is high partial pressure of carbon dioxide there. So the blood will become deoxygenated. So remember, in the alveoli, right, in the alveoli, reverse reaction will occur. Opposite to it will occur. Why? Because, okay, fine, in the tissue there is high partial pressure of CO2, right, low partial pressure of oxygen. So we know that CO2 will form this and that there, plus high partial pressure of CO2 will also form carbaminohemoglobin, all clear. Now, that deoxygenated blood, where will it go? It will go to the heart. From the heart, it will go to the lungs for its oxygenation. So when it will go to the lungs, what's going to happen? Basically, we want to exhale that CO2. We want to throw that CO2 out. So in the alveoli, what's going to happen? The reverse reaction. Matlab here in the alveoli, there will be reverse chloride shift. There will be reverse chloride shift, right? So in the tissue, basically your bicarbonate will diffuse out in plasma. It will diffuse out in plasma. Basically bicarbonate, it will diffuse out in plasma. Yes or no? Right? And chloride ion will enter RBC from plasma. So this direction should be clear, right? So this direction should be clear. So chloride will enter RBC from plasma, and that's how it is. Now here what's going to happen? Reverse chloride shift. The chloride ions in the RBC, they'll go out, and in response to that, bicarbonate will enter here. So when that bicarbonate will enter, they will bind with the H positive, right? Because here is low partial pressure of CO2, hemoglobin. Hemoglobinic acid will release that H positive. It will bind here. Carbonic acid will form. Then again it will break with the help of carbonic anhydrase, CO2 and water. So CO2 will go out. That is about the transport of oxygen. That is about the transport of oxygen. And overall, from all the chapters, never forget to revise. Never forget to revise the disorders and regulation part. Take disorders and regulation part. Take, so under normal condition, it is the 4 ml of CO2 that is delivered to alveoli by every 100 ml of blood. So please write down. So oxygen is 5 ml, and this one is 4 ml. Write it down. Up to this part, all clear? Is it clear? Okay, sure. Chaliye, let's move to the Next part, next topic, double circulation, right? Okay, ECG. Chale, ECG we will revise quickly. So next is double circulation. So what do you know about the double circulation? And we will quickly revise the cardiac cycle as well. So what do you know about the double circulation? Yes, please. What do you know about the double circulation? I believe I'm perfectly audible. Yes, I believe I'm perfectly audible. So what about the double circulation? That is my question right now. Yes, that is my question right now. And this is something very easy. It is not at all difficult. It is not at all difficult. Hanji, double circulation. See, bache. So you know that what kind of heart do we have? We have the closed circulatory system. So basically we discuss about the open circulatory system and closed. In the open, heart is there. There are few large blood vessels, but we do not have the blood vessels that are innervating each and every organ. This is what we know when we talk about the open circulatory system, like in the case of cockroach or arthropoda. Right now, in the closed circulatory system, heart is there, blood vessels are there that are innervating all the body organs. Now, when we talk about the circulation, right, let's talk about the single circulation first. So when you people talk about the single circulation, where will you see this? Where will you see this? Of course, in the case of fishes. Right. In their case, if you talk about the heart, that is two chambered. One auricle, one ventricle. One auricle, one ventricle. One auricle, one ventricle. What do we have here? One auricle, one ventricle. One auricle, one ventricle. Right, bache? What do we have here? One auricle, one ventricle will be there. One auricle and one ventricle will be there. So heart is two chambered here. Take here basically the heart is known as the venous heart. Why do we call it as the venous heart, bache? Because this heart will just pump deoxygenated blood. Right? It will not at all pump. It will not at all pump oxygenated blood. It is in the gills. The oxygenation will occur. It is in the gills that oxygenation will occur. Otherwise, it is not like that. Take care. So single circulation is there. Heart will only receive deoxygenated blood. Next word is incomplete double circulation. It's a simple topic, but yeah, question can come. Right? Incomplete double circulation. Incomplete double circulation. So that is what we are going to see in the case of amphibians. In the case of reptiles as well. In the case of reptiles as well. So in the amphibians, we have three-chambered heart, right? In the amphibians, we have three-chambered heart, right? In the reptiles, heart is mostly three-chambered, and here you can say that incompletely, yeah, partially, right? Incomplete, yeah, partially, yes, divided. Like in the ventricles, there is a septum that will partially, right, divide the ventricles, not completely. Is that clear? Is that clear? Is that clear? Right. So here in reptiles, you will see ventricles are incompletely. Ventricles are partially divided, not completely, partially divided. And in the case of reptiles also, we have one more thing that you need to know, that there are exceptions as well. In the reptiles, you will see the four-chambered heart. And if you remember, in the case of crocodile, right, bache, that's an exception. That's an important question. So in the case of amphibian and reptiles, let's suppose this is right auricle, left auricle, this is the ventricle, right? This is the ventricle. So if right side will have deoxygenated blood, left side will have oxygenated blood. In the ventricle, it will get mixed, right? So heart will pump mixed blood. So that's why incomplete double circulation. In the reptiles, this ventricle is partially divided. That's the difference. And in the reptiles, crocodile is the exception. Now, when you talk about the double circulation thing, what do we discuss? Double circulation. We'll practice the questions as well. Don't worry. Right? We will practice the questions as well. Right? I have added the questions. Right? Whatever topics I have mentioned accordingly, I have added the questions. Right? And we will discuss that too. Fine? We will discuss that too. Fine? Fine. So let's talk about the double circulation. Let's talk about the double circulation. So you have two auricles. You have two ventricles. You have two auricles. You have two ventricles. Right, bache? So right auricle is there, right ventricle is there, left auricle is there, left ventricle is there. Okay? So how many chambers do we have? Four-chambered heart, double circulation. Where do you see this? We will see it in the case of birds, in the case of mammals. So class apes and class mammalia, right? So basically what you need to understand, you know that right side, I told you about the trick. What is the trick? The trick is LOD. So left side will have oxygenated blood, left side will have oxygenated blood, and right side will have deoxygenated blood, right? Deoxygenated blood. So here's what's going to happen, right? So if right side is having deoxygenated blood, right, and left side is having oxygenated blood, so you know it very well. Right side has to carry that blood to the lungs. Why? For oxygenation. Right side has to carry the blood to the lungs for oxygenation. Who's going to do that? Pulmonary aorta. Pulmonary aorta. And after that, oxygenation. Your pulmonary veins, right? Your pulmonary veins, they'll bring back the oxygenated blood, because these are the exceptions I told you already, right? So these are the exceptions I told you already. It will bring back the blood to what? It will bring back the blood to the left auricle. It will bring back the blood to the left auricle. It will bring back the blood to the left auricle. Okay? So this blood will pass to the left ventricle. Then systemic aorta. Systemic aorta will carry it to the body parts, right? Body organs except lungs. Now from the lungs, you know that the veins, right? They will take it back to the right auricle. So here you will see two circuits are available, right? What is there in the case of the double circulation? We talk about the two circuits. Just say we have pulmonary circuit. Of course, if the word is pulmonary circuit, your lungs are involved. You have the systemic circuit where lungs are not involved. This is what double circulation is, right? This is what double circulation is, right? One side of the heart is having impure blood, other side of the heart is having the pure blood, right, bache? So basically, right portion is the pulmonary heart. It will form pulmonary circuit. Another is the systemic heart. It will form systemic circuit. So pulmonary circuit may ultimately you will see the blood, right. So basically what is going to happen here? So right ventricle will carry the blood to the lungs, and from the lungs that oxygenated blood will pass to the left auricle, right? This here left ventricle will carry that blood right to the body organs except, so lungs is not there, body organs except lungs, and then veins will carry it to the right auricle. Right, bache? So these are the circuits that we have. So just let's revise it from the NCERT. So simply they are saying that blood is pumped by right ventricle, enters the pulmonary artery, left one will be into aorta or systemic aorta. So deoxygenated blood is pumped into pulmonary artery, passed on to the lungs, from where the oxygenated blood is carried to the pulmonary veins into left atrium. So this is pulmonary circulation, as I said. Any doubt? Any doubt? Any doubt? Yes. Any doubt? Sure. Sure. Any doubt? Done? Sure. So this is the pulmonary circulation. Then oxygenated blood enters the aorta, is carried out by the network of arteries, arterioles and capillary to the tissue, where you know that deoxygenated blood will be collected by venules, veins and vena cava. Take, and that will pass to the right atrium. So this is the systemic circulation. This is the double circulation that we discussed. I hope this part is crystal clear to everybody. Right? I hope this part is crystal clear to everybody. Baki bache, you know that SA node, when you talk about the nodal tissue in the heart, you know that heart can start the wave of contraction by itself. So SA node is there, AV node is there, AV bundle is there, right? Basically, you can simply write, even if you want to write bundle of His, which will further divide into right and left, and then comes the Purkinje fibers. Please give it a quick revision. So, SA node, what is it? Sino-auricular node. Also known as natural pacemaker, natural pacemaker. AV node, pace setter, because it will delay the wave of contraction by 0.1 seconds. Right, bache? So SA node in the right auricle, in the right side of the right auricle, right? AV node is there in the right auricle, near to the, near to the AV auriculoventricular septum. Okay, from this AV node, like let's suppose if it is here, AV bundles will arise, that will be there in interventricular septum, that will divide into right and left, and then they'll give some short fibers, Purkinje fibers, right, that are there in the ventricular musculature, and then it will bring about the contraction, right? Contraction, we know it very well. So systole, diastole is there, right? Systole, diastole is there. What is there? Systole, diastole. Systole, diastole. So systole is the contraction. Systole is the contraction. Diastole is the relaxation. Diastole is the relaxation. So we have the contraction as well as the relaxation. Contraction as well as the relaxation. Systole is there, diastole is there. Keep it in your mind. Now, you know that joint diastole 0.4 seconds, where both auricles and ventricles they are relaxing, or auricular systole 0.1 second, ventricular systole is of 0.3 seconds. Keep it in your mind. Okay, keep it in your mind. Understood? Keep it in your mind. Sure, bache? Are you sure? Pakka? Any doubt? If there is any doubt, do let me know. If there is any doubt, do let me know. So auricles, if they contract for 0.1 seconds, right? So relaxation time will be auricular diastole will be 0.7 seconds. Here, right? Ventricular systole, ventricular relaxation, yeah, diastole will be for 0.5 seconds. So that's why we say the joint diastole is of 0.4 second. The joint diastole is of 0.4 seconds. So remember, right side, left side of the heart, bache. So right auricle, right ventricle, left auricle, left ventricle. Right, bache? Right, bache? Rest, right? So right side will have right T, tricuspid valve. Tricuspid valve. Left side will have mitral valve or bicuspid valve. Bicuspid valve. Now, at the base of pulmonary aorta, at the base of pulmonary aorta and systemic aorta, at the base of pulmonary aorta and systemic aorta, we have semilunar valves. What do we have? We have semilunar valves. What do we have? We have semilunar valves. Is it clear? Is it clear? Is it clear? Yes? Sure? Are you sure? So that's what you have to remember. So you know that here I'll quickly, and yes, do revise it from the NCERT that during auricular systole, which valves are open, which valves are closed, because they are important. So remember, when there is the closure of AV valves, that is auricular valves, auriculoventricular valves, that is your tricuspid and bicuspid valve. That is your tricuspid and bicuspid valve. That is what? Tricuspid or bicuspid valve. There is the first heart sound will form, that is love. First heart sound will form, that is love. So when there is the closure of semilunar valves, then what is going to happen? There will be the second heart sound, dub, right? Second heart sound, that will be dub. Okay, dub. Okay, okay. Chaliye, that is what you have to keep in your mind. That is what you have to keep in your mind. Is that clear? Is that clear? Is that clear? Sure? Take. So remember, this is the heart. So when bache there is the auricular atrial contraction, when there is the auricular contraction, AV valves are open, the blood will pass to the ventricles. When there is a ventricular contraction, when it will just start, so AV valves they are closed, but because of that pressure, semilunar valves will open and the blood will pass further, and when that ventricular pressure will come down, the semilunar valves will get closed. These are the kind of things you need to focus. Take, let's solve few PYQs, then let's start the muscles sliding filament theory, one of the most important topics. Take one of the most important topics. Take, so we'll switch also a bit, like now we are just doing human physiology. We'll also move to the anatomy part as well. Okay, anatomy part as well. So let's solve the questions from the mechanism of breathing as well. ECG also, yeah, ECG, Kwanisha, ECG, electrocardiogram, right? You know that when you are talking about the machine, when you are talking about the instrument, then it will be electrocardiograph, right? But the results that you will get, electrocardiogram. So P, Q, R, S, it is important. And it is, I believe ECG is the simplest topic. It is the simplest topic. Yes, do read it, right? But it is very easy. You have P, Q, R, S, T waves. You know that end of T waves means end of systole. QRS means ventricular contraction. So I believe this is the easiest topic that you guys can do. Take, chaliye, mechanism of breathing, answer the questions here. I hope these questions are clearly visible to all of you. Right? Kwanisha, do revise that, but. Right? Okay. What should be the answer, everybody? Please answer the question. Please answer the question. Answer the question. In which of the below two situations inspiration takes place? Intrapulmonary pressure remains less than the atmospheric pressure. There is negative pressure in the lungs. They go, inspiration. There is negative pressure in the lungs. That is for sure. Now they are saying intra, meaning within the lungs, the pressure remains less than the atmospheric pressure. So yes, tell me what should be the answer? What should be the answer? Very good. The answer will be 3 because both the statements are having the same meaning. Next, breathing involves dash during which atmospheric pressure is drawn in and by which the alveolar air is released out. So choose the option which fills the blanks correctly. Choose the options which fill the blank correctly. Hanji, which will fill the blank correctly. Do it. Yes, please do it. So breathing involves dash during which atmospheric air is drawn in. So it is the inspiration, and during expiration alveolar air is released out. Easy. Next question. Right. Next question. It's a statement based question. Please read it. Please read it. Hanji. So inspiration is initiated by the contraction of diaphragm, which increase the volume of thoracic chamber in the dorsoventral axis, or should it be anterior-posterior axis? Please tell me. Then contraction of external intercostal muscle lift up the ribs and sternum, increase in the volume of dorsoventral axis. So here Both the statements are incorrect because when diaphragm muscles they contract, the volume will increase towards anterior posterior axis, right? Anterior posterior axis, anterior posterior axis. What about the external intercostal muscles? In that case, dorsoventral axis. In that case, dorsoventral axis. That's what you have to keep in your mind. Next, right? Next, match list 1 with list 2 to find out the correct option. Here we have the list 1 and we have the list 2. And we have the list 2. We have the list 1, we have the list 2. So tell me please. Tell me please. Answer the questions. So, when you are talking about the inspiration, right? Expiration, external intercostal muscle, internal intercostal muscle, inspiration. So, what should be the answer? Expiration is exhalation. Inspiration is inhalation. What a simple question it is. External intercostal muscles, they will contract during normal inspiration and they will contract during forceful expiration. So, 1 with the C, 2 with the D, 3 will go with A. So fourth should be the correct answer. Next question. Choose the incorrect statement among the following. Choose the incorrect statement among the following. Quick. Choose the incorrect statement among the following. Yes, please. Incorrect statement. So forceful expiration is an active process. True. Mammals have negative pressure breathing. Acha. Internal intercostal muscle and abdominal muscles are the muscles of forceful inspiration. Acha ji. It should be inspiration. No. Respiration removes CO2 water. Of course. So third will be the correct answer. Why? Because here instead of inspiration, it should be expiration. That is why. Chale, let's move to the next question. Transport of gases. Hanji, answer this question. Answer this question. Today I told you na that the question practice will also be there in the session. So yes, please. Yes, please. %97% of the oxygen is transported as D. Low. You got the answer. And they all are NEET PYQs. Next question. Decarboxylation of carbaminohemoglobin takes place when? Decarboxylation of carbaminohemoglobin takes place when? Yes, please. So decarboxylation of carbaminohemoglobin takes place when? Yes. Decarboxylation. So of course, when the partial pressure CO2 will become low, right? Oh, sorry. When the partial low, right? And yes, that too in alveoli. So we cannot just read this statement. It should be in alveoli, and here the partial pressure of oxygen is high. Chali, next part. The process of migration of chloride ions from the plasma to RBC and bicarbonate ions from RBC to plasma is. RBC to plasma is. Then from RBC to plasma is. Then from RBC to plasma is. Yes, please. Exactly. Chloride shift. What is the another word? Hamburger phenomena. What is the another word? Hamburger phenomena. Right. Next question. Sorry. Assertion and reason. Hanji, answer it. Try to give me the answer here. CO2 diffuse in RBC and dissolve in water of cytoplasm to form carbonic acid. Hanji. Formation of carbonic acid occurs in the presence of carbonic anhydrase. True. But yes, reason is not explaining the assertion. Reason is not explaining the assertion. Simple. Now, the next question, statement-based question it is. You have the statement 1, you have the statement 2. So what are we supposed to do here? What are we supposed to do here? Yes, what are we supposed to do here? What are we supposed to do here? Yes. Yes. Hemoglobin is a red colored iron containing pigment present in RBC. True, oxygen can bind with hemoglobin in irreversible manner. It has to be reversible. It has to be reversible. So, 2, statement 1 correct, 2 is incorrect. Next, ECG. Yes, I didn't explain, but yeah, I'm going to ask the question from this particular part. So, answer the question. Answer the question. Answer the question quickly. ECG. Answer the question quickly. Quickly. Answer the question quickly. Hanji, what should be the answer? Please be quick. So read the following statement and find out the incorrect statement. For a detailed evaluation of the heart function, multiple leads are attached to the chest region. Okay. Take the end of the P wave marks the end of the systole. No. Just now I use this line that it is the end of T wave that marks the end of the systole. So of course this is our correct answer, but let's read other. The ventricular contraction starts shortly. The ventricles will start contracting shortly after Q and marks the beginning of systole. True. By counting the number of QRS complex that occur in the given time, we can tell the heartbeat. So is it clear? Is it clear? So simple type of questions will come from this part. Next part is again assertion and reason based question it is. Assertion and reason based topic it is. So read the, so end of the T wave marks the end of the ventricular systole. End of the T wave marks the end of the ventricular systole. Yes. 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 The end of the T wave marks the end of systole, yeah, end of just ventricular systole. So T wave represents the return of ventricle from excited to normal state, which is basically the repolarization. So basically the answer 1 is correct, where assertion and reason both are true, and R is correct explanation of assertion. R is correct explanation of assertion. R is correct explanation of assertion. Are we clear here? Yes, please. Any doubt? Sure. Any doubt? Yes, please. Okay. Let's move to the next question. So given below is ECG of a normal human. Which one of the following component is correctly interpreted below? Given below is the ECG of a normal human. Which one of the following component is correctly interpreted below? Yes. What should be the answer? What should be the answer? So, peak P, matlab when I read it, I was like peak P should be correct because it is the initiation of atrial contraction only. But I didn't focus the left word. If they have mentioned left atrial contraction only, definitely that cannot be the answer. Matlab, right? So, okay, P and R, they are representing systolic and diastolic. Is R representing diastolic? Not at all. Right? But complete QRS will show one complete pulse. That's true. That's true. That's true. Right, bache? Chaliye. Next topic. Again, related to ECG only. Right? Related to ECG only. So, yes, please. Yes, please. Yes, please. So, ECG is, electrocardiogram is the record of electrical activity of the heart which shows certain rates. ठीक? The electrocardiogram gives important information concerning the spread of excitation signal to the different part of heart, and it is of value the diagnosis of case of abnormal cardiac rhythm and myocardial damage. Is that true? Is that true? So what should be? Very good, bache. So both the statements are correct. Next, which of the following correctly explains a phase या event in cardiac cycle in a standard electrocardiogram? In a standard electrocardiogram. In a standard electrocardiogram. Tell me. In a standard electrocardiogram. Please tell me. So which of the following correctly explains a phase, or right, will explain a phase and the event in cardiac cycle in a standard electrocardiogram? Yes. QRS complex indicates atrial contraction. Nahi. Right. Time between anxiety represents atrial systole. Nahi. P wave indicates beginning of ventricular contraction. Nahi. So the answer should be 2, of course. Right. So now the next topic. Right. And then we will solve the question. So let's start the sliding filament theory. So you want the sliding filament theory first or the sections first? You want the sliding filament theory first or the sections first? Or the sections first? Or the sections first? So let's start. Okay, let's start. So before starting the sliding filament theory, let's talk about the muscles first. Okay, see, when you talk about the muscles, we know that, right? When you talk about the muscles, we know that they are mesodermal in origin. What they are mesodermal in origin. What they are mesodermal in origin. Yes, they are mesodermal in origin. Do we have the exception? Yes. Which exception do we have? We have the exception of iris muscles. Muscles of the iris. What is that iris? That colored part of our eyes. That is an exception. That muscles are ectodermal in origin. Now, here, see, we know that we have the skeletal muscles, smooth muscles, and chalo, let's start like this. Smooth muscles, cardiac muscles, and then what do we have? So we have smooth muscles, cardiac muscles, and we have the skeletal muscles. And we have the skeletal muscles. That is what we have, people. That is what we have. Smooth muscles, the structure is fusiform, spindle shaped, that is. Fusiform, spindle shaped, that is. Right? And it is going to be uninucleated. Uninucleated. And because it is smooth, why do we call it as smooth? Because there will be no striations, no lines, and they are involuntary. We cannot control them with our own will, and they are uninucleated. So, right, they are uninucleated. Understood? Pakka? Sure? Take. Right? Then we have the cardiac muscles. In the case of cardiac muscles, they are also involuntary. We cannot control them with our own will, but they are striated. They have the striations, right? They are branched. They are branched. Theek bache? Theek bache? In the case of cardiac muscles, the one word from where your question can come is that they have intercalated discs. But check cardiac muscles, right? Our heart is having these cardiac muscles. They need to work together as a unit. They need to work together as a unit. They have to contract all together. So these muscles, they are also fused, right? These muscles, they are also fused. So that junctions in between, we are using the word intercalated disc for it. What is the word that we are using for it? Bache, we are using the word intercalated disc for it. What is the word that we are using for it? Intercalated disc. Bache, break will be there at 10:30, right? Before that, you are not going to get any break, right? Kindly pay attention. Okay, kindly pay attention. Okay, sure. So the smooth muscles, right? The smooth muscles, cardiac muscles, and skeletal muscles. The smooth muscles, cardiac muscles, and the skeletal muscles. That's what we have. Up to this part, all clear? Sure. Up to this part, all clear? Sure. If there is any doubt, do let me know, please. If there is any doubt, do let me know. Do let me know. Just allow me 1 minute. Take, pakka. Okay. So, intercalated skeletal muscles, They are voluntary. We can control them with our own will. Are they multinucleated? Yes, bache, they are multinucleated. Okay, associated with skeletal system, right? And they are heavily striated. They have the striations, right? There are other points also. Right now I'm focused on these points. Now we have to discuss the ultrastructure of skeletal muscles. What we have to do, we have to discuss the ultrastructure of muscles. What we have to do, we have to discuss the ultrastructure of muscles. Is that clear? Is that clear? Is that clear? Skeletal. So in the case of muscles, let's suppose this is the muscle, right? Here you will see that muscle will have so many muscle bundles. So many muscle bundles are together making that muscle. So many muscle bundles are together making that muscle. So many muscle bundles are together making that muscle. So when you talk about the muscle bundle, right? When you talk about the muscle bundles, what do you discuss? When you talk about the muscle bundles, so they are also known as fascicles. They are also known as fascicles. And all these muscle bundles, just imagine this is one muscle bundle, this is another, right? There is one more, let's suppose, there is one more, let's suppose, there is one more, like this. When there are so many muscle bundles together, they are connected together with the connective tissue sheath, right? They are connected together with the connective tissue sheath, right? So connective tissue sheath that is connecting the muscle bundles, right? That is connecting these muscle bundles, which are also known as fascicles. I'm sticking to the NCERT only right now, right? This is known as fascia. This is known as fascia. This is known as fascia. Is that clear? Is that clear? Now in these muscle bundles, what you people will see? Now in these muscle bundles, what you people will see? You will see muscle fibers. What you people will see? You will see muscle fibers, right? And when you talk about the muscle fibers, these muscle fibers are anatomical unit of muscles. What are they, people? They are the anatomical unit of muscles, right? So what do we call it? Anatomical unit of muscle. So many muscle fibers are making the muscle bundles, right? So I'm repeating this part again. Just imagine you have so many muscle fibers. So many muscle fibers. These muscle fibers are together making muscle bundle. These muscle fibers are together making muscle bundle. What is the terminology that we are using for them? Right? We are calling them as fascicles. We are calling them as fascicles. Right, bache? We are calling them as fascicles. Is that clear? Is that clear? Is that clear? So connective tissue sheath that is connecting all these muscle bundles and fascicles. Connective tissue sheath that is connecting all that muscle bundles, right, are the fascicles. We use the word fascia for it. We use the word fascia for it. What is the term that we use for it, bache? We call it as fascia. We call it as fascia, right? Now, so many muscle bundles together, they are making the muscle. So this structure clear? This structure clear? Now, when you talk about the muscle fibers here, so as I said, this muscle fiber, it is the anatomical unit of muscle. It is the anatomical unit of muscle. What do we call it, people? We call it as anatomical unit of muscle. What do we call it, guys? Anatomical unit of muscle. Now here, this is the muscle fiber, right? The structure is not up to mark, but just I'm trying to explain you something. Now in the case of this muscle fiber, the outer covering here is known as sarcolemma. That's the term that we use, sarcolemma. Lemma mane covering. The cytoplasm here is known as sarcoplasm, right? It is known as sarcoplasm. It is known as sarcoplasm. Here in the sarcoplasm, you will see parallel myofibrils. You will see parallel myofibrils, right? There are so many parallel myofibrils. Actually, these parallel myofibrils, proteinaceous they are responsible for striations. Basically, when you look at these myofibrils, my dear students, when you look at these myofibrils, you will see the way actin myosin are placed. You people will see the light and the dark bands here, and they are responsible for the striations, right? They are responsible for the striations. And other than that, my dear students, Other than that, you know that sarcolemma done, sarcoplasm done. So sarcoplasmic reticulum is present in the muscle fiber. That is basically the storehouse of calcium. That is basically what? That is basically the storehouse of calcium. That is basically what? That is basically the storehouse of calcium. So you have sarcoplasm, having myofibrils, having sarcolemma, having sarcoplasmic reticulum, having sarcosomes as well. So this is about the muscle fiber. Now, even in the case of muscle fiber, when you look at the sarcolemma, you'll see at places it will show invagination. You'll see at places this sarcolemma will show invagination, right? Infolding. We call them as T-tubules. And adjacent to these T-tubules, on the either sides here, what you people will see? What you people will see? You will see this sarcoplasmic reticulum. Now, the nerve fibers, they'll innervate this. Just imagine this is a neuron, right? This is the nerve ending. So, bache, here you see a junction. What do you see? Here you see a junction. What do you see? A junction. What do we call it? Motor end plate or neuromuscular junction. Motor end plate or neuromuscular junction. What's the terminology that we use? Motor end plate or neuromuscular junction. That's the word. Motor end plate, neuromuscular junction. Motor end plate, neuromuscular junction. That's the word that we have. So you know that synaptic vesicles, they'll release the neurotransmitter, right? So here the muscle contraction, the wave of contraction will pass, right? So this is the motor end plate or neuromuscular junction. Yeah, motor junction it is. It is a junction in between the neuron and the muscle, right? In between this neuron and the muscles, we have the junction here. So obviously that wave of contraction will pass here. So what is going to happen? These endoplasmic reticulum, they will release the calcium ion and then the muscle contraction will start. So yes, we do need to discuss that part, right? We do need to discuss that part. But before that, we have to discuss the structure of sarcomere. Take. So here you guys can see, right? Tendon. What is this tendon? It connects bone to muscles. Right? It connects bone and muscles. Yeah, bone to muscles, yeah, muscles to bone, whatever you want to write. Overall muscle is having the connective tissue, right? Covering epimysium. Muscle bundle is having perimysium, endomysium. Do we need to remember? No. But do we need to remember fascia? Yes. Because fascia is the one that will join all the muscle bundles together. Now in the muscle fibers, right? Now in the muscle fibers, as I said, it is the anatomical unit of muscle. Now it is the muscle fiber, which is also known as anatomical unit of muscle. What is it? It is known as anatomical unit of muscle. It is known as anatomical unit of muscle. So what is happening here? You can see nerves are innervating it. It is having this structure. We are calling it as myofibrils. So when you talk about these myofibrils, they have something. What that something is, let's discuss that. Actin and myosin. Chaliye, let me draw that. After this part, I'll give you the break. Don't worry about it. After this part, I'll definitely give you the break, and that too for 15 minutes so that you guys can have your dinner, right? Even I have to eat something. Then after that, right people, after that, we will discuss it further. Just suppose that kind of structure. So when you look at that myofibril, you'll see, right? You'll see some bands are there. You'll see some bands are there, right? You'll see some alternate light and dark band. Alternate light and dark band. What do we see here? Alternate light and dark. Light, I band. Isotropic is the word here. Dark, arc, A band. And isotropic is the word here. So, in the light band, right, I. So which protein do we see? This contractile protein, actin. Right? There are other proteins also, but remember the actin here. In the dark band, that is the A band, you have, as per NCERT, you have myosin. But in our dark phase, our friends, they stay with us. Our well-wishers, not all the friends, but our well-wishers, they stay with us. So here you will see myosin along with that. Actin is also there. But yeah, as per NCERT, only myosin is there, mainly it is. So alternate dark and light band is there. Yeah, light and dark band is there, whatever you want to write. Now, in the I band, let's suppose this is your I band. A band. Because we just said that they are alternate. Chalo. right? A band, then again the I band is there. Now when you talk about this I band, right, from this I band, right, a zigzag kind of, right, from this I band you'll see something, we will call, matlab this I band will be bisected, right? This I band, it will be bisected. Yes or no? Yes or no? This I band, it will be bisected by Z line. It will be bisected by Z line. Take Z line is going to bisect it. So when we use the word bisect, matlab we are dividing it into two equal parts. We are dividing it into two equal parts. We are dividing it into two equal parts. Done, bache? Yes, please. Take, so it will be bisected, Z line is there. So this is that Z line, this is that Z line. Now here in the, yes, now here in the I band, A band, here in the A band, what you people will see? You people will see here in the A band also, there is a line that is crossing this. It is the elastic membrane actually holding it all together. You have M line. We'll discuss everything. Now when you look at the dark band, bache, this is the complete dark band. This is the complete dark band. So here in this dark band, that is A band, you will see that at places the actin is basically, actin is basically overlapping the myosin, and here you will also see that there is an area in between where myosin is not overlapped by actin. So it is the H zone. So H zone means Henson zone. H zone means Henson zone, right? So here myosin is not overlapped by. Here myosin is not overlapped by actin. What's the word that we have here? Myosin is not overlapped by actin. Myosin is not overlapped by actin. That's what you all need to keep in your mind, right? H zone. So overall you can see at the ends, right, the actin is also overlapping the myosin, but in H zone myosin is not overlapped by actin. Take, so when you look at these Z lines here, Z lines here, so between two successive Z lines, right, this is your sarcomere, functional unit of contraction. Functional unit of contraction. What's that? It is the functional unit of contraction. Just look at this. Here, dekho. I band will be bisected by your Z line or Z disc. Take, M line is also passing from the center of the dark band. Take this. So in between these two Z lines, right, between two successive, mane one after another, Z line, we have sarcomere. So it is the functional unit of muscle contraction, right? It is the functional unit of muscle contraction here. Functional unit of contraction it is, right? Right? Now this is the myosin. So myosin is also attached to the Z line. Here this protein is known as titin, not written in NCERT, but you can write it, right? This myosin is the thick band. So H zone is the area where myosin is not overlapped by actin, where it is not overlapped by actin, not overlapped by actin. So Z line is the functional unit of muscle contraction. Take, okay. So ye dekho, what is this Z line? You can just write it down, right? So It is an elastic fiber called Z line, which will bisect it. I told you it is the elastic fiber. This M line, what about this M line, bache? Yes, so thick filament in the A band are also held together in the middle of this band by a thin fibrous membrane called M line. Do not forget these points. Take, so when you talk about the sarcomere, that's how we can write that sarcomere means two half I band plus one A band. That's what sarcomere is. Two half I band plus one A band. That's what sarcomere is. Take, chale. So let's have a break. Then let's connect after 15 minutes. So it will be, let's meet at Hanji 10:50 p.m. So enjoy the break for 15 minutes. Fine. Enjoy the break for 15 minutes. Yes, please. Am I audible? So please let me know the structure of sarcomere. Is it clear to you or not? Z line, A band, I band, H zone, right? The area where myosin is not overlapped by actin. Is it clear to everybody? Is it clear to everybody? Because if it is clear to you, then the next part will be easy. And again, it is not given in NCERT, but yeah, the myosin will attach to the Z line here. And what do we call it? We call it as titin. What do we call it? We call it as titin. So I believe this portion is clear. Okay, done. So this is all about the sarcomere structure, M line. Now what we have to understand, we need to understand the structure of contractile protein. What we need to understand, people? We need to understand the structure of contractile protein. So whatever regarding that, you know, just let me know, please. Everybody here in the class, whatever regarding that, you know, please let me know. Please let me know. Yes, please. Please let me know. Anybody here in the class? Anybody here in the class? Structure of contractile protein. Yes. Structure of contractile protein. Structure of contractile protein. Do you know about it? Chaliye, let's start. So basically, when you are talking about the contractile protein, what is the meaning of this word? Obviously, the proteins which can undergo contraction, right? The proteins that can undergo contraction, right? The proteins that can undergo contraction. So here, firstly, let's talk. Firstly, let's talk about the actin. Right, firstly let's talk about what? Let's talk about the actin. So basically bache, again the word that we have used is ki it is a contractile protein. What's that? It is a contractile protein. Now here, right, we know that actin is forming the thin filament. That is basically, right, actin is forming what? Thin myofilament. Myo word is associated with muscle. So what that actin is making? It is making thin myofilament. It is making thin myofilament, right bache? Now in the case of this, you will see that it is not just the actin, other proteins are also there. But how are we going to start? We'll start with the G actin. G mane globular actin, right? It is monomeric, right? It is monomeric. What's that bache? It is monomeric. What's that bache? It is monomeric G actin. Now so many G actin, just say let's suppose these are the G actin, G actin, G actin, G actin, G actin, G actin, G actin. So many G actin, what will happen to them? They will polymerize. They will polymerize, right? So of course for the polymerization also they need the magnesium ion and they will form F actin. What will they form? They will form F actin, right? F here is fibrous, or you can also call it as filamentin actin, right? So globular actin is monomeric. It will polymerize, right? It will polymerize. And what will it form? And what will it form? Tell me. And what will it form? It will form filament actin. Now, these two filamentous actin, they'll be helically coiled like this. Like this. They'll be helically arranged like this. Right? They will be helically arranged. So imagine, this is your 1F filament. So 2F filament. That is how they'll be helically arranged. Right, bache? What? 2F filament, right? So globular actin will polymerize, it will form F actin. That F actin will do what? They will be helically bound, they'll be helically arranged like this. So remember, whenever you talk about the G actin, in the case of G actin, you will see that there will be myosin binding sites. What will be there? There will be myosin binding sites. What will be there? There will be myosin. Right, there will be on the G actin, there will be sites that can bind with myosin, So that is how it is. Okay, That is how it is. So this portion is clear. This portion is clear. Now we have to talk about the another protein there, that is troponin, right? Troponin. Matlab this troponin. This troponin will also cover this, right? They are also two. They will cover this F actin like this, right? It is going to cover the F actin like this, right? So basically, let's suppose if it is the F actin, you have the myosin binding sites over it. So this troponin is going to cover that. This troponin is going to cover that. Are you getting my point? This troponin is going to cover that, right? Because when the muscle is not contracting, right, these myosin binding site on the globular actin, on the filamentous actin are not allowed to bind with the myosin. So troponin is going to cover it. Now, at places you have one more protein, right? At places you have one more protein that is known as troponin. So this troponin is a trimeric protein. What kind of protein it is? It's a trimeric protein. What kind of protein it is? It is a trimeric protein. What? This troponin, right? It is having 3 polypeptide chains. It is having 3 polypeptide chains. So we have troponin, right? So it is troponin tick, T-I-C. Troponin tick, T-I-C. So basically troponin T, troponin I, troponin C. Right, so it will be the trimeric protein which will be present here, right? Which will be present here. So troponin T, I, and C. Let's suppose T, I, and C. So troponin T. See, troponin T. Of course, it will bind to tropomyosin and it will bind to 2 other subunits there. Troponin I. So it is, of course, bind to 2 other subunits, plus it will inhibit. It will not allow the actin. It will not allow the actin to bind with the myosin. Troponin C. This is the one that will bind with the calcium ion and it will initiate, right, that muscle contraction thing. That's the point here. Is that clear? Is that clear? So this is about the actin. Now, let's talk about the myosin. Myosin, which will form thick myofilament. Which will form thick myofilament. So we have thin myofilament and thick myofilament. Right? We have thin myofilament and what do we have? We have thick myofilament. So this part is clear. This part is clear. We have thin myofilament. We have thick myofilament. So here also you have meromyosin. What do you have here? You have meromyosin. What do you have here, bache? You have meromyosin. So meromyosin is again what? Monomeric form. So many meromyosin will join and they will form this. So in the meromyosin, what will you see? You will see HMM, you will see LMM. What is this HMM? HMM means heavy meromyosin, right? LMM means light meromyosin. It means light meromyosin. So this one is heavy meromyosin. This one is light meromyosin. So heavy meromyosin, it will contain globular head. It will contain globular head plus, yes, plus globular head plus short arm. Yeah, cross arm, globular head plus arm. Light meromyosin will have tail. So here in the heavy meromyosin, this globular head is an active ATPase. It can hydrolyze the ATP. So the structure is something like this. You all have seen it in the diagram, right? So this is the heavy meromyosin. This part is the light meromyosin. This part is the heavy meromyosin. So as I said, it will have globular head. It will have globular head and an arm, right? And an arm. So basically, one more better diagram of this part. This, take this. So here you will have actin binding sites and you will have ATP binding site. So what do you have here? So here you will have actin binding site. What do you have here? Here you will have actin binding site and here what do you have? You have ATP binding site. So it is the globular head that is an active ATPase. That's also a PYQ. That is also a PYQ. Right, Bache Gayatri? That is also a PYQ. So what's going to happen? These so many meromyosins, right? They'll polymerize. So this head and short arm, right? At regular intervals, they'll come out. Like if let's say this is the main axis, this globular head will be like this. Then next arm, globular head will be like this. So they will polymerize. So what do we call? So at regular intervals, that head, this cross arm will come out, right? So when there will be the signal for the muscle contraction, then what is going to happen? They will join. Chalo. So let's discuss that part. Right? Let's discuss that part. Okay, so each actin filament, right bache, each actin filament is made up of 2F actins. Each actin filament is made up of, yes bache, right? Each actin filament is made up of 2F actins helically bound to each other. 2F actins helically bound to each other. Each F actin is a polymer of monomeric G actin, that is globular. So two filaments of another protein. Tropomyosin also run close to F actin throughout the length, and troponin is distributed at regular interval. So in the resting state of the subunit of troponin covers the active binding site of myosin on the actin filament. Do we have any doubt here? Do we have any doubt here? Do we have any doubt here? Yes. Do we have any doubt here? Yes. Now, for the myosin also, I told you this thing. For the myosin also, I told you this thing, that each myosin or thick filament is also a polymerized protein. So many monomeric, that is meromyosin, that will form one thick filament. So they have globular head with short arm, that will form HMM. This can come in paper. When there is a short tail, then it is the heavy LMM, light meromyosin, right bache? So head and the short arm projects outward, right? Let's suppose this is the thing. The head and the short arm, they'll project outward, right? Then again, head and the short arm, they'll project outward at a, right, regular distance and an angle from each other. At a regular distance and an angle from each other from the surface of polymerized myosin filament. So it is known as the cross arm, right? Right, so this globular head and short arm, they will project outward at regular distance and angle from each other from the surface of polymerized myosin filament, and this is what we call as cross arm. This is what we call as cross arm. Understood? Sure. So globular head is an active ATPase. So the diagram should be clear. So here you have actin binding site, ATP binding site, the word cross arm is there, and here you have this actin thing. So are we clear here, or do you have any doubt? Yes, is it clear to everybody? Is it clear? Is it clear? Pakka, sure? Are you sure? Are you sure? Okay, chaliye, let's move to the next part, people. Let's move to the next part. So look at this, right? This is the myosin head. So it will bind to that, right? It has to bind to the actin for the contraction. So let's discuss the mechanism of muscle contraction. So when you talk about the mechanism of muscle contraction, you know that it will be explained by sliding filament theory, right? How can we explain it? We will explain it with the help of sliding filament theory. So here also, bache, we will discuss everything to the point only, right? Now you know that the nerve, right, the nerve cells, they'll innervate your muscle fiber. They'll innervate your muscle fiber. In a very simple language, I'm going to explain it. So when they innervate your muscle fiber, right, they will release the neurotransmitter. That neurotransmitter by T tubules, they will tell endoplasmic reticulum, that is your sarcoplasmic reticulum, to release the calcium ion. And bache, when the calcium is there in the cytoplasm, this calcium will bind with the troponin C. This calcium will bind with the troponin C. This calcium will bind with the troponin C. So once it will bind with the troponin C will slide, will move, right? Why? Because it has to unmask the sites, right, that can bind to the myosin head that are present on actin. That is why. So it is the sliding filament theory that will explain it. So here, let's read the points, bache. Before reading the points, let's revise it from this particular part. So it's a top 50 concept. There should be the quick revision. So that's how we are going to revise it. After the sliding filament theory, let's have a quick revision of glycolysis. Krebs cycle. Chalo, I'll do it. Plant growth regulators, Z scheme, and then the sections and all, right, and endocrine glands and all, mechanism of hormone action. So everything we will cover today. Okay, so see students, many times you people say that the teachers, they have started the series, they are not finishing it. The reason is you people are not even focused. Even you guys, you don't attend the lectures regularly, or if you do, you just keep on chatting. Okay, so these kind of things are there that also demotivate the teachers, that if they are not interested in, you know, learning that chapter, that what are we supposed to do? Okay, what are we supposed to do? So if you really want us to continue this part, if you really want us to, you know, discuss these important topics in detail, then please be consistent. Okay, it's a request, people, please be consistent. Okay, chaliye, so let's pay attention here. So nerve impulse, they arrive, right? Nerve impulse, they arrive at the axon terminal. Nerve impulse, they arrive at the axon terminal of the motor neuron. It will trigger the release of neurotransmitter. So which example we have discussed here? Acetylcholine. Which example we have discussed here? It is acetylcholine. It is acetylcholine. So see this nerve impulse, acetylcholine will be released, bind with the muscle fibers. So as this acetylcholine, bache, it will diffuse. As it will diffuse across the cleft, bache, it will bind to the receptor in the motor end plate. Motor end plate, basically we are talking about the junction here. We are talking about the neuromuscular junction here. Is that clear? Is that clear? We are talking about the neuromuscular junction here. Is that clear? We are talking about the neuromuscular junction here. Understood? Sure? Sure. So neuromuscular junction, then of course the action potential, the wave of contraction basically will be there in the muscle. Now, after that, what's going to happen, right? Obviously, when one neurotransmitter will be released here, it will pass the action potential to the respective cell. And after that, the enzymes are there in this gap, bache, that will destroy that particular neurotransmitter. And that will be again reabsorbed by this previous neuron so that they can resynthesize it. So same as the thing which is written here, ki acetylcholine esterase. Because here we are talking about acetylcholine. So that's why the enzyme here is acetylcholine esterase. In synaptic cleft, it will also destroy ACH. So another muscle action potential doesn't arise unless more acetylcholine is released from the motor neuron, right? Right. So next muscle contraction will start, right? Next muscle contraction, the wave for next muscle contraction will start when, the next time the neurotransmitter will be released. Now, after that, what's going to happen? Once, like from the sarcoplasmic reticulum, calcium ions will come out. Sarcoplasmic reticulum is having the channels, right? They are having the calcium channel. So calcium will be released and that will be there in the sarcoplasm and that will bind to the troponin. That will bind to the troponin. Where will it bind, bache? It will bind to the troponin, which is there on the thin filament, right? Which is there on the thin filament and which will expose the binding site for myosin. Imagine it like this. Suppose this is the troponin, right? This is the troponin. Here you know that this is the actin filament. Imagine this is the actin filament. Actin filament is further covered with actin filament is having the binding site that are further covered with tropomyosin. Over that we have the troponin. So when this troponin will get the calcium ion, let's suppose these are the calcium ion. When this troponin will bind to the calcium ion, when they will bind here, so they will slide. They'll move this troponin. So when they will move this troponin, the tropomyosin will also, right, will also move, and the sites on the actin which can bind with the myosin, they will get exposed, right? They will get exposed. So that is the complete thing here. Okay, so calcium release channel in the sarcoplasmic reticulum, they'll close after that, right? It's not like that sarcoplasmic reticulum will keep on releasing the calcium, right? It will keep on releasing the calcium, right? It's not like that. Right, later on they will close. So calcium active transport pump used ATP to restore the low. So basically that's a simple thing. Calcium release channels in the sarcoplasmic reticulum, they will close, and there are some, so these calcium ions actively they will transported. They'll transport it. Right, bache? Done. Then troponin tropomyosin complex will also slide back into the position where it blocks the myosin binding sites on actin. Initially, right, when that sites will be away, the myosin head will bind to the actin, right? And then what is going to happen next? Yes, bache, what is going to happen next? So see, firstly, so after calcium bind to the troponin, I told you contraction will occur. right? For contraction, this is the power stroke. It will use ATP. Do we need ATP for the contraction? Yes, we do need that. Now, myosin head has to bind to the actin, right? And then, bache, what will it do? When myosin head will bind to the actin filament, right, it will bring it towards the center, towards that M line. So basically the actin filaments will slide over the myosin. This line is very important, right? It is the actin filament that will slide over the myosin. It is the actin filament that will slide over the myosin. I'm repeating this part again. It is the actin filament that will slide over the myosin. Am I clear here? Yes. Am I clear here? Yes. It is the actin filament that will slide over the myosin. Right, bache? Right. And after that, calcium ion will be resequestered. So you can get an idea from this particular picture as well. So myosin head, right, they will bind to the actin and they'll, right, bring it towards the center. Okay. So this is the diagram that you need to keep in your mind. Okay, this is the diagram that you need to understand, right? So troponin at places, tropomyosin at places, troponin is there. So when calcium ion will bind to the troponin, right, it will slide. It will also slide the tropomyosin. So these sites will get exposed. These sites will get exposed. Is that clear? These sites, they will get exposed. Now, ATP is required. We know that globular head is an active ATPase. It will hydrolyze the ATP. So this point is also very important. Remember, when ATP is attached to myosin head, then it is not forming the cross arm. Then it is not attached to the actin. It is not attached to the actin, right? So you can read it here, कि ATP is hydrolyzed when myosin head is unattached. Matlab, when myosin head is not attached to the actin, then here ATP will bind. So ATP has to be hydrolyzed then. So ATP is hydrolyzed when myosin head is unattached. This is important. This is a PYQ. And yes, students, they made the mistake there. Then ADP plus inorganic phosphate, when it is attached to the myosin head will attach to the actin. That's how they form the cross bridge. Take. So when to this head ADP and inorganic phosphate is attached, then they will bind with the actin. So this is what we call as cross bridge. Is that clear? This is what we call as cross bridge. Is that clear? Is that clear? Sure? Are you sure? Are you sure? Pakka? Pakka? Done? Are you sure? So next is ADP and inorganic phosphate. ADP and inorganic phosphate, they'll release. So when they release, they'll cause the head to change the position, and that is why actin filament will move, right? So when ADP and inorganic phosphate, when they release, it will cause the head to change its position. So they'll bring that actin filament close to the center, right? But so binding of ATP will cause the myosin head to return to the resting position. Again, I'm saying that's a PYQ. This is important. This diagram is also very important. Okay. Okay. Sure? Are you sure? Are you sure? Pakka? Chaliye, let's move to the next part. Then see, these are the diagrams from the NCERT. So if you people are having any doubt here, do let me know. If you people are having any doubt, do let me know. If you guys are having any doubt, do let me know. So the diagram part, this part is important. Rest. There are, ye dekho. These are the different diagrams which are showing this. So rest. These are the 6 points that you need to remember. So of course, bache, when there is a muscle contraction, the sarcomere, right? The size of the sarcomere will contract. If it was this, so when it will contract, even its size will also decrease, right? So whenever there is the muscle contraction, ultimately you have seen the protein fibers are present. Ultimately what we have seen, protein fibers are present. Just say I band is there, A band is there, I band is there, A band is there, I band is there, A band is there. Is that clear? Is that clear? So what do we have? I band is there, A band is there. I band is there, A band is there. So ultimately here the length of I band will get reduced. But we are not concerned with, we are not saying the length of actin and myosin will reduce. It's simple. It's simple. Actin filament, they were placed here. They were placed here. When there is the muscle contraction, they'll come closer. They'll come closer, right? So overall, if the length of I band was this, now this will be the length of I band. So length of I band will decrease. Length of I band will decrease, but not the length of complete actin filament. If, for an example, if actin filament is of 5 centimeter, it will be for 5 centimeter only. It's just that, let's suppose actin filament was placed like this. Now there is a muscle contraction. Here you have the another actin filament, so they'll come closer. So length of I band will get reduced because basically myosin head will pull it towards the center, right? But the length of actin filament, if it was of 5 centimeter, it will be 5 centimeter only. Right, it will be 5 centimeter only. If it was 2 centimeter, it will be 2 centimeter only. So it is the length of I band that will get, right? It is the length of I band that will get reduced, but the length of A band will remain same. But the length of A band will remain same. Right, so it is the length of I band that will get reduced, right, bache? But it is the length of A band that will remain same. But it is the length of A band that will remain same. Right, it is the length of I band that will get reduced, but it is the length of A band that will remain same. So obviously length of sarcomere will decrease, length of myofibril will decrease, and Henson zone disappear. So this is also PYQ, this is also PYQ. That which bands their length change, which band their length will remain same. So do revise it, right? So length of actin and myosin filament will remain same. Have a quick revision of it, quick. Just have a quick revision of it. Just have a quick revision of it. Yes. Done? Any doubt? Any doubt? So this is all about the sliding filament theory. Do revise it, right? Do revise these things. Lactic acid fermentation, you know na, when anaerobic breakdown of glycogen is there, lactic acid will form. So Cory's cycle that will operate in between the liver and the muscle, right? That will basically change that lactic acid again. So I will not discuss that, but right, you have to do it by yourself. And if you have any confusion, you can just check the Game of NEET series, bache. You can check the end game series. In that we have completed that particular chapter in detail. Right, we have completed the chapters in detail there. Okay, chali. So next topic that we need to start is glycolysis. That is of course from what? That is of course from your respiration part. What is the next topic that we have to start? We have to start glycolysis. I'll stick to the important things only, right? So if you know about this, very good. If you don't know it, just listen to me now. So glycolysis, glyco, the word is glucose here, the word is breakdown here. The word is glucose here, the word is breakdown here. So it's a 10 steps process. It's a 10 steps process, and oxygen independent it is, right? Oxygen independent it is, and it is there in the cytoplasm. Cytoplasm, oxygen independent it is, and 10-step process it is. ठीक, right? So बच्चे, whenever we talk about the respiration, whenever we talk about the respiration, in the case of respiration, we have one thing, right? And what is that? We have to break carbon-carbon bond. You know it very well. So oxygen is going to oxidize the glucose, the energy will be released. This is what we know in general. Now, you talk, whatever you discuss, You can consider the example of anaerobic organism. You can consider the example of aerobic organisms. Whatever you want to consider, you people can. You can take the example of anaerobic organism. You can take the example of aerobic organisms. Whatever you want to discuss. देखो, basically the topics that we discuss, be it glycolysis, Krebs cycle, it is basically the cellular respiration at the level of cell. Right, at the level of cell, when we discuss this respiration thing, we say that the breakdown of glucose is there. Now here, what is the point that I want to mention? Right, so whenever you talk about the aerobic cell or the anaerobic cell or anaerobic, these are the two words. Aerobic cell and anaerobic, these are the two words. Aerobic, having that need oxygen, no requirement of oxygen here. Right, no requirement of oxygen here. And bache, if I talk about the evolution, if I talk about what? If I talk about the evolution, if I talk about what? If I talk about the evolution, right? So you know that anaerobic organisms, anaerobic cells, they came first, and then aerobic cells, they came. So when you talk about the respiration, and when you talk about the first step of respiration here, it's same. It is your breakdown of glucose only. It is the glycolysis only. Right? It is the glycolysis only. So first step of respiration that we discuss is glycolysis. Right? As I said, 10-step process. So glucose. What is this glucose? It is C6H12O6, 6 carbon containing compound. So in 10 steps, right? Overall respiration is a multi-step process, and each step is enzymatically controlled. Each step is enzymatically controlled. So it's a multi-step process, enzymatically controlled it is. So glucose will break into 3 carbon containing compounds, which we call as pyruvate. So one glucose will give you 2 pyruvate, 2 pyruvic acid. That's what we discuss. How many steps are there? 10 steps. So what we have to remember, देखो, there are few things that you need to remember, or few reactions. First thing, how many steps are reversible or irreversible? How many steps are reversible or irreversible? Steps in which ATP used or formed. Steps in which substrate level phosphorylation occur. Understood? Sure? Done? So this is what we need to discuss. So let's start the discussion. It is from my favorite book, Strayer Biochemistry. So, right, this is what you can follow, because in NCERT also many points are not clear. So basically we start with the glucose, and bache, we say that this step is irreversible. Like firstly, let me mention the information here. Right, the flow chart is in front of you, and trust me, it's simple. Glucose will give you glucose 6-phosphate, will give you fructose 6-phosphate, fructose 1,6-bis-phosphate. That will give you glyceraldehyde 3-phosphate and dihydroxyacetone dihydroxyacetone phosphate. So dihydroxyacetone phosphate will also form glyceraldehyde 3-phosphate. So you will have 2 glyceraldehyde 3-phosphate, then 1,3-bis-phosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate, and pyruvate. That's it. Trust me, it's simple. It's simple. Right? It's literally simple. You just give it a try. You just give it a try. Okay? You just give it a try. Trust me, you are going to love this topic, even the Krebs cycle. For the Krebs cycle, right, we have the tricks as well. What do we have? We have the tricks as well. Understood? Understood? Take, chale. So, Okay. So let's discuss this part. Right, let's discuss this part. How many steps are irreversible? 1, 3, 7 and 10. 1, 3, 7 and 10. Remaining all are reversible. Substrate level phosphorylation. 2 steps only. Which one? 7th and 10th. Do remember these reaction. Do remember these reactions. Take, it's important. Now, the first step, the glucose 6-phosphate. Basically, bache, as I said, what is it? It's a 10-step process. What is it? It's a 10-step process. So because it is a 10-step process, 5 steps here are considered as preparatory. 5 steps here are considered as payoff phase. 5 steps may, it will be preparatory phase. It will be preparatory phase. It will be payoff phase. So in the preparatory phase, basically ATP used, here ATP synthesized. ATP used, here ATP synthesized. Is that clear? Is that clear? Take, so the first step is the locking step here, right? So when you talk about the first step here, what is this first step? What is this first step? It is the locking step, right? See, glucose will be converted to glucose 6-phosphate. Basically, we are adding the phosphate. ATP will be used here. Remember, for each step, there are 10 different enzymes and magnesium ion is required. It is the cofactor here, okay? And when it comes to the regulatory enzyme, this one is also, there is one more, but you have to remember this. I'll give you the reason here. These are the regulatory enzymes they are. So glucose to glucose 6-phosphate. But this particular step is also known as the locking step. Why is it so? See, glucose can cross the cell membrane. It can cross the plasma membrane. It can go out. It can go out. But when it comes to glucose 6-phosphate, it is impermeable, right? It cannot cross the plasma membrane. It cannot cross the plasma membrane. So once, before the MSc entrance examination, right, one book was there. In that one book, it was the question that let's suppose a person is sick. When we are sick, when we are not well, doctors, they give us the saline solution, they give us this glucose. If in simple terms, if I have to say, glucose solutions will be given to you, saline solution is given to you, right? Basically, solutions are given to you. So why can't they directly give us glucose 6-phosphate? That was the question. Why can't they directly give us glucose 6-phosphate? That was the question, right? Why they give us the glucose? Because this first, right, because this first step here, the ATP is used. Body is already weak. Body is using its ATP. So why can't we directly give glucose 6-phosphate? So because it is impermeable. It cannot cross the plasma membrane. So that's why it is the locking step as well. So that is why here in this particular step, what exactly do we need? Yeah, what exactly do we want here in this particular step? Bache, we do not want. Yes, what we have to do here, right, we basically need to trap this glucose here within the cell. So glucose to glucose 6-phosphate will form. Glucose to glucose 6-phosphate will form. So it is irreversible. What's that? It is irreversible. So glucose, then there is glucose 6-phosphate, ATP will form ADP, magnesium ion will be used here. So what's the word here? It is hexokinase. Then glucose 6-phosphate, fructose 6-phosphate, these are the isomers. So enzyme used will be isomerase, right? So it is phosphohexose isomerase. So hexokinase, sometimes glucokinase is also written. Basic function is same. Whenever the word is kinase, phosphorylation will be there, right? It is related to the phosphate. It is related to the phosphate. But yes, there is a difference, right? There is a difference of affinity in between hexokinase and glucokinase, and that is your homework. And I want you people to answer the question in the comment section, right? Do check it and please let me know, right? Do check it and please let me know. Chali. Next, fructose 6-phosphate, fructose 1,6-bisphosphate. See, fructose 6-phosphate and fructose 1,6-bisphosphate. Now, there is a difference when you say diphosphate and bisphosphate. There is a difference, right? Diphosphate and bisphosphate there is a difference. What is there? There is a difference. But when it is a diphosphate, meaning 2-phosphate, they are attached to the same carbon only. I'm just writing P, I didn't mention the phosphate, just P, right? They are attached to the same carbon only. But when you are saying bisphosphate, bis is also 2, but these phosphates, they'll be attached to 2 different carbon. If you can see this image, right? If you can look at this structure, if you can look at this particular structure, so you will see this is the fructose, this is the first carbon where the phosphate is attached, this is the sixth carbon where the phosphate is attached. So that phosphate is attached to two different carbons. So that is why the word is fructose 1,6-bisphosphate. That's why the word is fructose 1,6-bisphosphate. That is why the word here is fructose 1,6-bisphosphate. Clear? Clear? Are you sure? That is why the word is fructose 1,6-bisphosphate. So up to this part, all clear? Yes, up to this part, all clear? Are you sure? Are you sure? Pakka? Pakka? Chaliye, so now, here in the first step, ATP will be used. In the third step, ATP will be used. Do mention the reaction. They are very simple. Glucose to glucose 6-phosphate. Right. Fructose 6-phosphate to fructose 1,6-bisphosphate. So basically one extra phosphate has been added there. Now, when it is the fructose 1,6-bisphosphate, 1, 2, 3, 4, 5, 6, 6 carbon containing compound. Now, bache, right, the cleavage will occur. Right. So when it is the cleavage, the enzyme used is aldolase. The enzyme used is aldolase. So it will give us, of course, it's a 3 carbon containing compound. It will give us something having 3 carbons, 3 carbons. And this one is the 5th step. This one is 4th step. This one is the 5th step. So basically it will give us what? Glyceraldehyde 3-phosphate, or we can also write it like this, PGAR, phosphoglyceraldehyde. Either you can call it phosphoglyceraldehyde or glyceraldehyde 3-phosphate, or glyceraldehyde 3-phosphate, or glyceraldehyde 3-phosphate. That's the word. That's the word. That's the word. And here you have DHAP, that is dihydroxyacetone phosphate. Dihydroxyacetone phosphate. Take. Right, so again here the triose phosphate isomerase is the enzyme. But here what is the point? This DHAP, it will immediately get converted into glyceraldehyde 3-phosphate. So no doubt when we break this fructose 1,6-bisphosphate, right, when we break this fructose 1,6-bisphosphate, no doubt we have the glyceraldehyde 3-phosphate and dihydroxy, right, dihydroxyacetone phosphate, right, bache. But basically what is the point? What is the point here? Right, what is the point here? The point here is that this dihydroxyacetone will get converted into glyceraldehyde 3-phosphate. Okay. Okay. Sure. Sure. Done. Done, bache? Sure. So at the end, what will we get? We will have 2G3P, right? Because dihydroxyacetone, it will also get converted into G3P. So we will have 2G3P. We will have 2G3P. We will have 2G3P. That's what we need to keep in our mind, right? So here, dekho, individual charts are also there. Take. So here you people can see, right? Irreversible step. This is also regulatory. This is also regulatory. Pyruvate kinase is also regulatory. Now this particular regulatory enzyme, main regulatory enzyme, but it is having, it is allostatic enzyme. If there is high concentration of ATP, yes, citrate, etc., right, its activity will be inhibited. Means how can we say that inhibited? That this is the regulatory enzyme, because this particular enzyme, my dear students, it will inhibit its activity if there is high ATP, right? So even the ATP, AMP, citrate, all of them, they affect its activity, but high ATP can. So if there is high ATP, right, this enzyme will not be able to act further. So basically the point here is the glycolysis will not occur, because ultimately it is not required. Right? It is not required. Done, bache? Chali. Then comes the fructose 1,6-bisphosphate, dihydroxyacetone phosphate, glyceraldehyde 3-phosphate, done and dusted. Now, as I said, how many glyceraldehyde 3-phosphate? Later on you will get 2. Later on you will get 2. Now this is the payoff phase where we will see ATP will form. So glyceraldehyde 3-phosphate. Now how many glyceraldehyde 3-phosphate do we have? 2. So here the oxidation and phosphorylation, or in some books dehydrogenation is written. Oxidation bhi hoga, loss of electrons, and addition of phosphate is also there. So glyceraldehyde 3-phosphate will give 1,3-bisphosphoglycerate. It is aldehyde. It is glycerate, या glyceric acid, acidic group. Plus here 1 phosphate, here 1 more phosphate. So again, are we going to use the ATP here? No. Here what will happen, bache? Inorganic phosphate will get added. Inorganic phosphate will be added to this glyceraldehyde 3-phosphate plus this NAD, nicotinamide adenine dinucleotide, bache. It is oxidized. It will be reduced. Right? So why do we write 2 pi here? 2 pi matlab inorganic phosphate here, or 2 NADH here? Because we know that we have 2 glyceraldehyde 3-phosphate. Because we know that we have 2 glyceraldehyde 3-phosphate. Because we know that we have 2 glyceraldehyde 3-phosphate. Is that clear? Is that clear? Yes, bache, phosphofructokinase is a regulatory enzyme. Its concentration will, its activity will get inhibited, या get affected by the change in the concentration of ATP, AMP and all of this, and citrate, etc. ठीक? Chaliye, so 1,3-bisphosphoglycerate will form 3-phosphoglycerate. So this is the substrate level phosphorylation, an important, or I would say very important question. Dekho bache, see, phosphorylation. You know that what is the meaning? Do you know what is the meaning of phosphorylation? It's simple. Addition of phosphate. It's simple. Addition of phosphate. That is what phosphorylation is. Addition of phosphate. That is what phosphorylation is. Now it is substrate level phosphorylation. Other than that, we'll also read photophosphorylation. We will also read oxidative phosphorylation. So substrate level. So actually here the phosphate will be added by substrate. Just for an example, 1,3-bisphosphoglycerate. What do you have here? 1,3-bisphosphoglycerate. What do we have here? 1,3-bisphosphoglycerate. Right. Then it will give us 3-phosphoglycerate. Matlab 1 phosphate is less. So who's going to accept that? Adenosine diphosphate. It will become ATP. So this is the first ATP forming reaction here. Substrate level phosphorylation here. The question can directly come from it. Which enzyme is going to catalyze it? Phosphoglycerate kinase. Phosphoglycerate kinase. Right, जैसे hexokinase, pyruvate kinase, phosphofructokinase are the regulatory enzymes, right? Majorly phosphofructokinase, please keep it in your mind. So that is what going to happen here. Then 3-phosphoglycerate will form 2-phosphoglycerate. This 3-phosphoglycerate will form 2-phosphoglycerate. This 3-phosphoglycerate will form 2-phosphoglycerate. Is that clear? Sure? Are you sure? It will form this. Any doubt? Any doubt? Any doubt? So 3-phosphoglycerate or 2-phosphoglycerate, right? Then there is just the rearrangement. So for that type of steps, the reaction, the enzyme will be mutase. देखो phosphoglycerate mutase, phosphoglycerate mutase, understood? Phosphoglycerate mutase. Then बच्चे 2-phosphoglycerate will form phosphoenol pyruvate. 2-phosphoglycerate will form phosphoenol pyruvate, and here also the water will come out, and then the last step, phosphoenol pyruvate will form pyruvate. Then again it is the substrate level phosphorylation. Understood? Understood? Just revise it quickly. Just revise it quickly, and if there is any doubt, do let me know. So basically, right, basically in 2 steps, ATP used, then revise it. Right. So basically 2 ATPs used, but how many ATPs formed? 4 ATP ATPs formed and 2 NADH2 formed, and these NADH2 बच्चे in electron transport chain, they will also give us ATP. So if I talk about the actual concept, 1 NADH will give us 1 NADH2 will give us 2.5 ATP. So in the NCERT still 3 ATP is given, but yeah, as per actual concept it should be 2.5 ATP. So now out of these 4 ATPs, see 2 ATPs are used now. Matlab it's just like you took 10,000 rupees from me and you are like, madam, I will receive 30,000, then I will return your 10,000. So net gain will be 20 in your case now. Let's say today you ask me for the money. You are like, give me 10,000 rupees. Okay, I gave you. Then after 10 days, you receive 30,000 rupees. But your net gain will be 20 only because you took the 10,000 from me and you have to return it. Same is the case here. Right, bache? Same is the case here. So 4 ATPs, they were formed. Out of that, 2 ATPs, they were used. So 4 minus 2 will be 2 only. So the net gain is of 2 ATP and 2 NADH2. So if we go as per the NCERT, in electron transport chain, 1 NADH2 will give you 3 ATP. So 2 will give you 6, right? 2 NADH2 will give you 6 ATP. So 6 ATP from NADH2 plus 2 ATP that already form total 8. So if you solve the PYQs, you'll get some answers in which it is written that from the glycolysis in totality, you will get 8 ATPs. Right? But actually 1 NADH2 will give you 2.5 ATP. Accordingly, we have to answer it with the accurate concept. So let's have a quick revision of this part. And then let's move to the question practice sessions. Take. Then let's move to the question practice session. Heartbeat is there. Chale, you do it. Revise it. So done? Yes? Is it done? Are you sure? Narayana, there is a separate video on this channel where I have explained the plant life cycles of the plant, so you can revise it from there. Chaliye, let's solve the question. Answer this question. Answer this question. Mechanism of muscle contraction is best explained by. Easy question. Very easy question. Do it quickly. Beta, even I am tired. Do it quickly. Do it quickly. Yes, please. It is the simple question. It's a very simple question. Chaliye, let's go. So it is the sliding filament theory. Next, sarcoplasmic reticulums are abundant. Acha. For red muscles, okay. Myoglobin content is high. Sarcoplasmic reticulums are moderate. They are aerobic muscles, depends on anaerobic respiration for energy and less myoglobin content. So I'll tell you how to answer such kind of questions, right? I will even tell you that how to answer such kind of question. Dekho, if it is the red muscle enough, or you will say the myoglobin content will be high here. ठीक, मतलब they are aerobic. So if they are aerobic, they cannot be anaerobic. So fifth option is not given. Okay, already. Then, so they are aerobic muscle. Myoglobin content is high. So what about the sarcoplasmic reticulum? What about the sarcoplasmic reticulum? Are they abundant or are they moderate? It's a very important point. It is a very important point. And trust me, it's simple as well. It's simple as well. What should be the answer? What should be the answer? What should be the answer? Yes? Yes? Guys, are you sure it's one? Are you sure it's one? Are you sure? You have to tell the correct information. You have to tell the correct information here. You're talking about the red muscle fibers. So what should be the answer here? Right? Red muscle fibers. What should be the answer? We are getting different, right, answers. I want one answer. Yes, please. Beta, simple hai. Dekho, in their case, red myoglobin is more, even they have M for myoglobin, high mitochondria. Sarcoplasmic reticulum, it's low. So answer here, more accurate will be the fourth one. Next, assertion and reason type of question. Masking of actin filaments. Masking of actin filaments take place during the muscle relaxation. Acha. Mg ions are pumped back to the sarcoplasmic cistern. देखो, magnesium ion directly incorrect है. What about the A1? So masking of actin filament take place during muscle relaxation. Yes. Yes, बच्चे मिथुन. What should be the answer? Either the chat is slow. I don't know what is the issue. Right? Why everybody is not answering the question? So obviously here this is correct, but this is incorrect. Third has to be the answer. Next, what would happen if ATP suddenly were not available after the sarcomere had started to shorten? Acha. If ATP will not be available, no ATP matlab? No ATP matlab? But ATP, no ATP, if ATP will not attach to that head part, you know that what will not happen? If they are not available. So don't you think the answer should be 1 and 2? Right? Because when you talk about the ATP, what that ATP will do? When that ATP will attach to the myosin head, right, that myosin head will detach from actin. So binds to the troponin. Sorry. Okay, sorry. So cross bridge would not be able to detach from actin. Right? Muscles will be there in the state of rigidity. They are not relaxed. They'll be contracted. So answer is fourth here. Next question. Chaliye, next question. Yes, please. Next question. So, calcium is important in skeletal muscle contraction because it binds to troponin to remove the masking site, activate the myosin ATPase, prevent the formation of bond between myosin crossbridge and actin, detaches the myosin head from actin. So, you know that one will be the correct answer. ठीक? So, thyroid gland, we need to discuss it yet. And glycolysis. So bache, this is all about today. Let's discuss it up to this part only. Right bache, rest, because there will be 4 parts of each. Right, 4 parts for physics, for chemistry, for biology. Right, so let's discuss it in the next class. Okay, so next class may we'll revise the Krebs cycle, right, the Z scheme, plant growth regulators, and so on. And we will pick up the topics from the class 12th as well, class 11 topics and class 12th as well, right? So let's complete it up to this part only. Okay, so I will definitely share this PDF in the Telegram group, but moreover, there is one more important thing that you all need to understand. See, and that is, see, this is our channel. You can go to any video, right? You can go to any video. You'll get the detail of the batches which are starting, right? Which are about to start, and the batches in which, right, which are about to start, and the batches, right, which you can enroll now, right? You will get the details basically. So if you will watch this particular video, here in this particular video, right, we'll see that how and the difficulty level of CBSE biology paper, how does it impact the NEET, number 1. Secondly, bache, this session, you can watch it. You can go to the description box of this particular video also. So basically, there is, for the NEET 2025 aspirants, right? For the NEET as a 2025 aspirant, there is a test series. So what you have to do, you go to the any video on our channel, you'll find the link for this test series, right, bache? So when you click on that, right, you can join our NCERT Crux test series, and the test will start from 27th of March. The initial 4 tests are part test, and then there will be the full syllabus test. Okay, so that is how, right now this link is not opening, but if you will click it, you'll get to know about it, right? Moreover, the new batches for the NEET 2026 and 2027, they are also started. So till 27th of March, bache, this is the benefit for all of you, that you will get the plus platform at just ₹4499. Right, you have to use the coupon code Ambika10, and you people will get plus platform at just ₹4499. What is this plus platform? The platform for your success, right, bache? If you enroll, right, if you enroll with us, then you people will get the access of all the batches that are running across the Unacademy platform, that are running on Unacademy platform for NEET UG category, right? Let's suppose you join my batch. I am going to start Nimbus English batch for class 11, Nexus English batch for class 12, and Phoenix, right, batch for the droppers, right? And 1st April onwards, these batches will start. So let me tell you, bache, if you enroll yourself, right, for the plus platform, and that too at this price, bache, you will have the access of all the batches. Let's suppose if you have joined Nimbus English batch, but let's suppose you want to change your batch, or you're okay with you want to study the physics from somebody else, chemistry from somebody else, you will get the chance to do that. Moreover, you will have the free access of test series. After every 15 days, there will be the test. You will get the PDF notes. You will be able to access all the content which is available on Unacademy platform, right? The soft copy of that. But if you want the hard copy, then you have to go for the iconic. So if you want more detail about the batches, do let me know in the comment section here, right? And do revise these topics. They are very important. And I'll share the PDF in our official Telegram group, that is Unacademy NEET English. So make sure you join that Telegram group as well. Okay, done. So do revise it, be sincere, and bache, on this channel I have posted the video in which we have discussed the 45 chapters that can give you 650 marks. So if you are struggling with the stable syllabus completion and the revision, if you don't know which chapters you should start first, you can watch that video. So till then, take care. If you like the session, do let me know in the comment section. Thank you so much, guys. Thank you, everybody.